Thank you very much for the kind invitation. I will try to be brief and to tell you these are the 3 slides that I have to present. So my talk will deal with the immunogenicity issue for biological and biotechnological products. And I want to go through again, through the concept, what is a biological, biotechnological product. I guess we have already, and we had already the opportunity to listen a lot of things concerning this type of products. But what I would like to underline in this presentation, at the beginning of this presentation, is that when we talk about biologicals and biotechnological products, we should think in terms of immunogenicity, according to, in my view, the 3 following concepts. The biotech product we have prepared can be a copy of a naturally occurring molecule, which is normally present in the human body, and we have several examples: erythropoietin, interferons, and other molecules, like also antibodies, humanized, full humanized antibodies. They can be, on the other hand, foreign molecules, which means molecules which do not exist in the human body. They are created, like fusion proteins, and they have no counterparts naturally available in humans. And the third issue is that the third case is that the recombinant product from the immunogenicity point of view can be a molecule which has been intentionally designed to be immunogenic. If I want to have a recombinant vaccine, I need to have the vaccine immunogenic, otherwise it doesn't work. So this is some clarification I would like to make, because according to the molecule we are thinking about, the issue of immunogenicity may be different. I will not touch the third point, which means vaccines, for example, because it's something that is not in my intention to discuss. Immunogenicity is a negative, unwanted phenomenon, and of course, the number of attempts that has been made during the development of a new product to reduce immunogenicity are very many, and they are often linked to the design of the preparation of the molecule itself, beginning from the preparation of the plasmid to the engineering approach, genetic engineering approach that it is followed. The first point I would like to make is that when a biological or biotechnological product is administered to animals or to humans, you may get neutralizing or non-neutralizing antibodies, and this will be further discussed later on by myself, but also during the presentation which will follow this presentation. Of course, in the clinical setting, careful planning of immunogenicity evaluation should include data systematically collected from a sufficient number of patients. I'm not going to enter in this business now, but the size of the study to understand whether a product is immunogenic and to which extent it is immunogenic may vary from case to case. And finally, what I guess we need to stress is the concept that for these products, immunogenicity is a never-ending story, and it has to be somehow further addressed in the risk management plan, which accompanies all the submissions which are related to this product at the European Medicines Agency. The consequence of an immune reaction to a therapeutic product may be transient or may be severe, and may also be of clinical significance or not, and this is something that we have to be taking into account. Moreover, you know that the immune system has a strong individual background. It is the immune response is coded by genes, of course, and according to the genotype of the patient, according to the disease, and according to a number of other aspects, the immune response can be quite different from case to case. Depending on the immunogenicity, or the immunogenic potential of the therapeutic product and the rarity of the disease, again, we may need a huge number or a large number of patient, of subject, to understand exactly how immunogenic is the product. Well, we have now some experience. The first recombinant protein was a monoclonal antibody. Actually, it was not a recombinant, it was a fusion protein. We are going back to 1980s. And now we have a lot of experience. At that time, the monoclonal antibody was the murine monoclonal antibodies, so nothing to do with humans. Was obtained from mouse and was produced in mouse, from mouse ascites, so in a very dirty way. And from them, we have now a lot of progress made. And for example, we now have recombinant antibodies, as you know, and monoclonal obtained from fusion fusion approaches that are not anymore available on the market. So we have decided in early at the CHMP level to set up in around 2008 a guideline on monoclonal antibodies and on immunogenicity. And a number of important aspects have been extended have been included in this guideline, whose scope is not restricted to recombinant product. And I would like to add that this immunogenicity guideline, which we will talk about, does not cover only biosimilars, for example. I heard on some occasions that the immunogenicity issue is an issue for biosimilar product. This is not correct. It is an issue for all recombinant biological product or even non-recombinant biological products. So this is a clarification I think has to be made. Therefore, according to this guideline, the evaluation of immunogenicity should be a multidisciplinary approach, taking into account all the possible expertise available in terms of quality, non-clinical and clinical. The guideline which is mentioned here has two general sections. Essentially, one is a main section, and the other one is an annex to the guideline in which you have essentially some technical details. I don't want to get into the details, but again, one important concept which has been already stressed yesterday, which is included in this guideline, is that you have to approach the immunogenicity issue also when you are running a comparability exercise. We heard a lot about the comparability exercise, which is performed within the same manufacturing process or between products when I want to prove a biosimilarity at the quality clinical and non-clinical level. Of course, the comparability exercise must include the immunogenicity evaluation of the product. Again, before or after changes made in the manufacturing process, or between an innovator and a biosimilar or potential biosimilar product. In the general section of the guideline, there are listed all these general concepts which are basic for the interpretation of the immunogenicity problem. Well, there is a general approach which is quite important in my opinion, dealing with factors which may influence the development of an immune response against a therapeutic protein. And again, in the guideline, there are a few concepts which are stressed. There are patients and disease-related factors, and you see some examples listed here, and I don't want to go into the details. And there are also product-related risk factors which somehow influence immunogenicity. And you have here protein structure, formulation, and aggregation, for example. It is well known that macromolecules are more immunogenic, for example, when they aggregate, unless they reach the higher level of aggregation in which they may become tolerogenic, but normally aggregates are more immunogenic than monomers, for example. And this is something which is quite important, for example, for monoclonals. In the general section, there are also some some points related to the non-clinical assessment of immunogenicity. We will hear again a lot concerning this aspect, and we have already heard a lot concerning this aspect yesterday and late today. What I would like to underline, to stress, is that the animal model is always a model. Of course, we may play around with the transgenic mouse, with immunodeficient mouse or SCID mouse. The real problem is that when we have to deal with a recombinant protein, which is a human protein, for example, and we inject and we treat animals with this human protein, we will get for sure an immune response. So we have to take into account, when you plan to use animal models, exactly what we are looking for and the various situations we may encounter, especially because, as I said, the molecules are not self for the mouse and for the rabbit or for other animals. The only exception which sometimes works, but which implies a lot of other issues, is the use of non-human primates, as you have already heard yesterday. And these have some implications concerning the ethical point of view. There is a big attention in the guideline to the assays for detecting and measuring immune response in humans. And I guess that this will be also treated in more detail in the following presentation. But of course, the assay strategy and the technical strategy to be followed to detect the antibody is very, very important. We must think that we are working with a large population, because we have to measure these antibodies, to look for these antibodies in the patients that belong to the clinical trial area. And so we have several patients to be screened, and we must have a method which is easy, which is reliable, and which is fast, maybe. And all these three things, they are not always easily done together. They are not easily developed in a sort of combined way, because often when you have a screening method, you may have some false positive or some false negative results. So it depends from case to case, but the process of developing the antibody assay is quite important. We will come back later on to the concept of neutralizing assays and things like that. Anyway, the characterization, the strategy to detect these antibodies and the strategy to characterize the antibodies is very important. Of course, why we are looking for antibodies and why we are talking about immunogenicity, because the consequences of immunogenicity are quite important, quite obvious, I would say. But just to keep it short, of course, the availability, the bioavailability of the product is quite fundamental, of course. And if I have antibodies, doesn't matter whether they are neutralizing or not, they may interfere with the distribution of the product in the body, and at the end it may interfere with the activity in that the antibody has to display. And therefore we must pay quite a great attention to understand whether we may have some influence of the antibodies we raise in humans in terms of efficacy. And of course, immunogenicity can also impair the safety of the product, because as you know, patients who develop antibodies may have some more frequent reactions linked to the infusion step of the administration of the product. And there are also cases of immediate hypersensitivity. There are a lot of other issues. What I would like to stress is the last point. Maybe we will hear a little bit more later on from clinicians and from other experts. The cross-reactivity with endogenous counterparts. If we administer a molecule which may be somehow already present in the body, and we induce with the administration of this molecule antibodies, these antibodies will react potentially with the native molecule which is present naturally in the human body. So this is something that has to be taken into account. Of course, the immunogenicity assessment should be part of the clinical trials, but of course it has to be also something which is already planned. I mean, the investigation and the avoidance of immunogenicity should be part of the development plan, of the clinical development plan, but also the development plan for the molecule itself. When I design a molecule, I have to take into account mainly the potential immunogenicity of this molecule, and I have to try to do all the best to avoid this immunogenicity. At the clinical level, of course, the study design has to be clearly defined to assess for the immunogenicity at the clinical level. There are, of course, influences from antibodies raised by the product at the level of pharmacokinetic, again the concept of neutralizing, but also non-neutralizing antibodies. To interact with the molecule, I don't need to have an antibody capable of directly neutralizing the activity of the molecule itself. I can simply have an antibody clearing away the relevant molecule from the blood, for example, and this would lead to a reduction of its activity and quite significant reduction in some cases of its activity. As I have already mentioned, it is quite important from this product and for this type of issues to take into account immunogenicity in the risk management plan. According to the European approach and to the European legislation, the applicant should present a risk management plan in accordance to the CHMP, in accordance with our European legislation and according to the guideline on risk management plan. The extent of data on immunogenicity that can be obtained during the clinical development program of a biotechnology derived product I'm reading, it's not as big as it should be. As I have already said, it depends from the frequency of the immune response against the given molecule that I can understand how many patients I have to treat before I can get formal and statistically significant information. So when I plan the study during the marketing authorization application, I have by default a limited number of patients. But then in the risk management plan, I have to explain to the authorities how and for how many subject and in which way I will continue to measure the induction of antibodies in the population which will be treated by the product. So in the risk management plan, I guess that all these things are quite important and should be better and properly detailed in order to let us know the planning of the company, of the applicant. As I said, there is an annex to the guideline in which there are a lot of details, in my opinion, on the types of antibody assay. I have already mentioned that from the technical point of view, it will not be easy to detect in a very reliable and validated way these antibodies, and especially because we have several different issues which have to be taken into account. I have to detect the antibody, I have to confirm it's present, I have to analyze the specificity of the antibody, its neutralizing properties if they are, and also other characteristics which may be important, and I have to take into account the fact that I need validated method, as I have already said. This is a table taken from the guideline in which a flowchart is reported, and it explains how to go through the results that I get when I test these SIHA from the various patients I have treated. I would like not to go into details, I guess that time is running and my time is over, but there is also a product-specific guideline dealing with immunogenicity, and it takes into account a special category of product, which are monoclonal antibodies, and of course, why we have already this guideline available? Well, because these, in my opinion, these antibodies are, the monoclonal antibodies are an emerging, strongly emerging market in the sense that in the pipeline there are a number of monoclonals which have been manufactured and authorized at the EMA level. There are a few monoclonals which will come as a biosimilar. So it's a very active sector in the pipeline of the applications to the agency, and this was the reason why it was decided to set this guideline a few years ago. And there are a few general statements. I don't want to go into details. You will have the slides. Of course, they are macromolecules, so there are no very specific issues we should think about. But we have to think that in the monoclonal antibody area, we have, as we heard yesterday, modified antibodies, conjugated antibodies. We have also fragments which may be used. We have nanobodies, mini bodies. There are a huge number of variables available in which the original antibody molecule is reduced or modified using genetic engineering techniques. So this means that we might have some unwanted immunogenicity, although the sequence of the structure of the antibody will be a fully humanized sequence, as it is the case nowadays. Again, the detection of monoclonal antibodies is not easy, so there is a section in which the guideline tells you something more how to deal with this specific aspect. And again, there is an immunogenicity risk management plan also for monoclonal antibodies. Risk identification is important, not only for antibodies, but in this guideline it was underlined this concept. And especially, again, when you design the molecule, when you assess before manufacturing the molecule, and you are also thinking about the mechanism of action, and there are a number of things that you may take into account, and the risk identification is quite important. So as a conclusion, what I would like to underline is that immunogenicity is a common issue for macromolecules, including biologics, and including biotechnological product, and including biosimilar, as I would stress. The mechanisms involved are very complex, and it is not easy to understand exactly which mechanism is used when, or is followed by the immune system when to mount an immune response. Trials available at the time of the marketing authorization application may be not large enough to identify less frequent cases. So this is the reason why we have a risk management plan. And this is also the reason why the risk management plan must take into account all potential issues linked with immunogenicity. I think that this was my last slide, so I thank you very much for your attention, and I have passed the floor to the chairman. Thank you very much.
Can hear me. I want to thank the organizing committee for inviting me and express my disappointment in not being able to be in Moscow. It looks like a very important and exciting meeting. And what I'd like to accomplish today is to present some of the major challenges facing biosimilars in regards to immunogenicity and approaches to meet these challenges. Next slide. I think one of the major concepts is that all protein products have some level of immunogenicity. That incidence may be extremely low, less than 1 in 2,000, or, as in the case of administration of foreign proteins, can be quite high and approach almost 100% response in patients. But importantly, I think one of the important concepts is that major changes in product manufacturing processes can have a major adverse impact on the immunogenicity profile of the product. And there's frequently no clear-cut root cause for this change. So as illustrated in the third bullet point on this slide, although there are multiple theoretical causes of immunogenicity, there are very few definitive examples of why a product is immunogenic. And whether it's published or not, we really don't have a full understanding of what's causing immunogenicity in the majority of products. Now, yes, we understand that foreign proteins are going to be highly potent immunogens when they're introduced into people. And we understand that aggregates of large sizes that start to look like viruses with repetitive array of proteins, these large protein aggregates can induce an immune response. But we don't know actually at what level how many of those particles you need to induce an immune response. So there's a lot of information that we really don't understand about protein immunogenicity. So the next slide, number 3, talks about generalized protein domains, just because I'll be using this type of figure throughout the talk. So in the solid green, you see non-receptor binding functional domain, so it's a non-functional domain, and the receptor binding domain in the triangle with the striped green lines. The next slide, on slide 4, we show an illustration of one of the impacts to product quality and clinical performance is the effect of antibodies on proteins. These antibodies can bind a non-functional domain, which would usually be non-neutralizing antibodies. They won't stop the antibody from performing its function. But if the anti-drug antibody binds to the functional domain, they can be neutralizing antibodies and prevent the biological function. Now, I was about to talk about what the impact of product quality can be in clinical performance on these products, but since we just heard a talk on the potential impact of unwanted immunogenicity on product safety and efficacy, rather than go through and repeat what's already been said, I was going to skip now to slide number 9. I'm moving my thing to slide number 9 to talk about what several of the regulatory regions have to say about immunogenicity in published guidance. And the first guidance I'd like to talk about is the EMA, where they published several guidances on monoclonal antibodies and biosimilars and immunogenicity guidelines. And I think there's 2 major points that I'd like to cover in the guideline. And the first one is that as you can see in the first bullet, the comparative assessment of unwanted immune responses against a biosimilar reference monoclonal antibody are normally undertaken as part of the clinical studies. So they're requesting head-to-head studies. The question is, why are head-to-head studies being requested? And there's really three answers to that, or three components. First, comparative analytical assessment will not provide sufficient assurance that the adverse impacts product quality, just because it's hard to predict what attribute will affect immunogenicity. Secondly, the immunogenicity in animals are not really predictive of immunogenicity in humans, so that you can't use animal studies to predict what will go on in the clinic. And finally, the testing results that you get in any immunogenicity testing is highly dependent on the methods and the study design, and you can't compare across different studies. And I'll talk more about this in a little while. The other part that I'd like to talk about in the guidance is the fact that when you're looking at and designing the immunogenicity studies, the guidance suggests that you take a risk-based approach, and that they offer some of the different types of risks that you should look at, but they really want you to define the risk in a very rigorous manner. And as for a risk-based approach, applicants really need to define what the risk is in a formal risk assessment. Now, on the next slide, you see that FDA recently published a biosimilar guideline, and in fact, it sort of mimics the same things that the EMA guideline says. It asks for head-to-head studies of at least one clinical study, and it's asking that the extent and timing of the clinical study will depend upon the range of factors, including the extent of analytical similarity and the incidence and clinical consequences of immune response. So again, this is a risk-based approach to what type of data the FDA wants to see up front and what will be post-approval in a pharmacovigilance program. So what is a risk-based approach? And that is on slide 11 now. When we talk about a risk-based approach, risk has been defined as a probability, that is how many patients are likely to mount an immune response in this case, versus the severity. What happens to a patient if they mount an immune response? And we've already heard about some of the very adverse events that can happen. And when you do these risk assessments, severity outweighs the probability of a risk. There is very little tolerance for a severe adverse event. If life-threatening events occur, those occur at very low level and are not tolerated. And you can see that by the evidence of a product being recalled recently where there were 3 deaths due to anaphylaxis, and the particular product was recalled off the market until that issue has been straightened out. So there is a real big outlay of probability of risks. Severity, when there's really a harm, there's a high intolerance for that risk. However, for most of the products that we talked about, the risks are pretty well known, and most events are not so serious. So you really do need an overall risk score, and that depends on an assessment of various factors that influence immunogenicity. On slide 12, I'll go through some of those things in terms of probability. There are some considerations looking at the probability. There's likely lower probability of several types of categories. If the patients are immunosuppressed, they're less likely to mount an immune response than patients that are being treated for autoimmune disease where there might be a heightened immune system. Single dosing, likewise, is lower probability than chronic dosing because you sometimes need to prime the immune system. More human is less immunogenic than foreign proteins. IV administration is much less immunogenic than sub-Q. And then highly pure tend to be less immunogenic than impure products, and partly because we think that's due to adjuvant effects. When you have all these other molecules, you might have an indirect stimulation of the immune system. And then no aggregates is, again, a probability that we see low probability of an immune response versus aggregates. But in aggregates, the real risk seems to be in large protein aggregates that show a repetitive array, sort of start to look like a virus. And the immune systems are highly geared to attack viruses. At least that is the theory. On the next slide, the other part of the equation, of course, is severity. And we have a fairly good handle on the types of things and what the severity risk can be. Likely to be more severe if you have an endogenous version of the product, well, you might inactivate that. If that endogenous version has a unique activity, well, you might actually wipe out that activity and cause major issues to the problem. This occurred with thrombopoietin, where anti-drug antibodies knocked that out and people became thrombocytopenic. So those are severe adverse events. If it's a sole therapy, you don't want to have neutralizing antibodies and have no other therapies to go to. If you have other therapies to turn to, that's less of a risk to the patients. Of course, life-threatening disease, if you get a neutralizing response to a life-threatening disease, that's not as bad as having a non-life-threatening disease and immune response and inactivity. And of course, there's other things on this list. I think the one that I like to talk about is anaphylactic responses, of course, for somebody that is a high risk as well. And that you can tell partly by what the innovator product has, but you still have to control that risk. The next slide, slide number 14, shows examples of anti-drug antibodies, incidence, and clinical impact. And the point of this slide really is to say that the rates of antibodies, either whether they occur at a high frequency or low frequency, do not correlate with the clinical impact. As you see with the high rate for human alpha-galactosidase, there's a high incidence of antibodies, and yet the clinical impact does not appear to be significant. In contrast, there are low rates of antibodies for thrombopoietin, either 0.5 to 4%, but neutralizing antibodies and thrombocytopenia resulted from some of these things. So even though it was a low incidence, you saw a clinical effect. Turning to slide 15 then, what are the considerations for analytical testing? So how do you really make these risk assessments into actual practice? And this slide table provides some examples of high score considerations and examples of low score considerations. When you have a high score consideration, you really want to be testing samples in real time. You want to get the data as soon as you can. You want to use very sensitive assays early in development. You want to take a more conservative approach to setting cut points so that you actually are sure that you're capturing all the potential false negatives. You want to test the positive samples for neutralization using a well-developed neutralization assay and do that in real time. In contrast, when the risk to product quality is lower, you can lay off on and take not such an aggressive approach. You can batch test at the end of the study sometimes, develop increasing sensitive assays during development, and just take a little bit more time in developing the product. Now, biosimilars, there is also a crunch that the timelines for analytical and some of these method developments are crunched more. So we'll have to see how much that actually impacts on the clinical developments for some of these things. And turning to another issue is really the immunogenicity testing. And one of the major points on slide 16 is that the rate of immunogenicity detected in an assay is wholly dependent on the assay chosen, its controls, the execution, and the clinical study design. But biosimilar companies are unlikely to know the actual details of all these assays. They may know something about what the type of assay was used and some of these parameters, but they won't know sufficient details to actually mimic and reproduce that assay exactly. And the problem is that these assays will then give relative information that will be different. Now, the optimal screening assay, if one had one, would be sensitive enough to detect any level of specific antibody present in patient serum, would have no false negatives, would have very few false positives, so you don't have to keep on confirming whether they're positive or not. You would discriminate between pre-existing antibodies and treatment-induced antibodies and detect antibodies in the presence of drug with a highly reproducible assay. Now, this is the ideal screening assay and actually doesn't exist, at least at this point. On the next slide, slide number 18, I'm presenting several of the common assays that are used to assess immunogenicity rates. And the first, in the upper corner, we have an ELISA, where you bound the drug to a surface, then expose it to the anti-drug antibody, which in this case would be the serum. You wash out, you add a detecting antibody that will bind to the initial antibody, which is usually conjugated with some marker, in this case an enzyme. You can do a reagent that get a color, and so that you can quantitate the amount of detecting antibody that's binding. And a similar type of assay is a bridging ELISA, where you have the drug conjugated to a surface, but in this case, when you add the serum antibodies, it's added to such a way that it will only bind to one of the molecules, and then you can add the conjugated drug to the other bispecific antibody, and you can actually then use a detection system. The third assay that I'm describing here is a radioimmunoprecipitation assay, where you radiolabel the drug, you add the serum sample, you let it incubate for a while, you precipitate the antibody-bound drug, and then you measure the radioactivity in the pellet. Now, all that's good, but on slide 19, it turns out that each assay has its own implications and own limitations for the results it reports. Now, ELISAs do have several issues related to design, and especially that the nonspecific background can be an issue, and when you stick the antigen to a solid surface, or in this case the product, that you might change conformation, you might have some different conformational epitopes not being exposed, so you can potentially miss some antibodies, anti-drug antibodies. It also has extensive wash and incubation times, which don't allow for sufficient binding or will eliminate low-affinity antibodies. And that would be fine if low-affinity antibodies were not important, but unfortunately, low-affinity antibodies have been correlated with some of these severe adverse events, including neutralization of endogenous activities. So these are molecules and antibodies that we really do need to track, but may not track by using these methods. So on slide 20, I think, again, each assay has its implications. And the bridging format appears to be less background. It does not require the use of an anti-IgA reagent, which makes it easier to do the validation. But again, you still have a problem with the loss of low-affinity antibodies. The radioimmune precipitation assays actually tend to be more sensitive format as compared to ELISAs, but they also have the problem that you may not precipitate all complexes. It's very dependent on the actual way you're precipitating, so you don't get necessarily all isotypes in some classes as you might get in some of the other methods. So again, it has limitations. The actual assay of choice is depicted on slide 21, which is surface plasmid residence. In this technique, a light source of polarized light is shined onto a sensor and looks at the interface between the gold film and the water surface of where the product is bound to dextrin, and then you see the gold circles or yellow circles, which represent the anti-drug antibody, can bind to it. When you have those proteins binding to the drug, there's a shift in the reflective index, and you will see an increase in the light, reflective light, and so you get a resonance signal. Now, the advantage to this particular method on slide 22 is that this involves exposure of a drug in a lawn of dextran that does not really affect the structure of the product, so the drug conformation seems to be correct. It's what presumably the body will be exposed to. It also is a real-time procedure, and therefore you can see a fast detection of antibody binding, and in fact you can look at the association, so you get both on and off rates. And because you don't have extensive washing steps, that you can actually see and detect low affinity antibodies, and you can actually see what the affinity for each antibody is, and you can measure that. So the fallback on this, though, is that the equipment is pretty expensive, needs expert knowledge and design, and to run the analyzer thing. And the throughput analysis, actually, although it's timely, it takes takes a bit of, it has a low throughput, so it takes a lot of runs to get a lot of samples done. So there are a couple of issues that have a dramatic effect on what the reported rates of immunogenicity are. And I've selected a couple of them to provide a little bit of more detail. And one of them is the assay cut point. And this is simply, at what point are you going to cause a particular optical density reading in some of these assays as a positive, and at what point do you say it's a negative? And typically this is done by taking a lot of negative samples and running a diagram. So as you see here, take the average and establishing a cut point, typically at 95% negative, so that you have a low level of false positives, but you try to eliminate as much as possible the false negatives. And you can see this happen. But you can obviously see that if you set the cut point differently, you're going to have a different impact on the actual rate of the seroconversion that you're reporting. If you set a high cut point, you're not going to see any antibodies that may be important. On the next slide is actually the other issue that can actually interfere with the reports, is that the circulating therapeutic protein can interfere with the antibody detection assays. This is illustrated here. If you have the antibody, the anti-drug antibody already bound to a drug, then it's not going to bind to the drug coated on the plate, and you're going to miss it. Whether it's bound to functional areas or to the non-functional areas, it's going to underreport. Now, the typical way to avoid that is to try to take sampling at times when there's low levels of drug in the patient's serum, so that you don't have this effect. But it is important to make sure that the impact of the drug in the sample screening assay is meaningful. And in this case, on slide 25, I show a figure where you are adding different amounts of the drug to the serum samples that has a tighter limit of detection of around 2. And you can see that as you add more and more drugs, suddenly there's a drop-off in the titer, meaning that the drug is being competitive against the sample that you have, indicating that there's some specificity, that this is, in fact, a positive test. Now, the other thing that agencies have requested is that identification of other immune classes. When there's certain skin autoimmune disorders that are known to involve IgE, when there's hypersensitivity reactions detected, agencies will typically ask for additional assays, IgE assays, things like that. And the reason is that the authorities really want to recommend that you elucidate the underlying immunological mechanisms for adverse events. That would facilitate development of strategies to mitigate that risk of critical significance. So if you know that an IgE event is involved, you might either measure patients, see the IgE response, and know further treatment for that, or develop some treatment regimens that will lower the risk to the patient. The other assays that are sometimes requested are either looking at histamine in patient serum or histamine release assays, again, to confirm anti-drug IgE and try to understand the underlying mechanisms. And here you see in figure slide 27, where you have an IgE receptor transfected cell. You add the anti-serum to it and you may, if there's IgE present, it can load the cells. You add the drug, you cross-links, and you can get histamine release. And that provides some idea about the mechanism of what you're observing in terms of the adverse events. Now, the final assay that I want to talk about is the neutralizing assay for antibodies, because it's any positive result when you get should be actually analyzed for the neutralizing capacity. And those can be analyzed typically in a biological assay, but sometimes you can have alternative binding assays. They may be appropriate, particularly when you use them as a potency assay for release. Now, I'm not saying that you use the exact assays for release. The assays that you use for neutralization should be developed particularly with an intended purpose of looking at neutralizing assays. So they're seldom going to be the same as you use as release assays, but it can be the same type of binding assay. If you think that's a biologically relevant assay to measure potency, there's no reason why you couldn't use those type of assays. Now, the problem, or one of the issues, is that you may see neutralizing activity, but there's always a question of what does that mean in terms of in vivo clinical impact. And frequently, people see these neutralizing assays, and there's no clinical impact. But one of the ways to look at that is when you do identify patients that have a neutralizing response, you can do subset analysis and look to see with those patients with a neutralizing response, do you see anything for PK or PD? Frequently, you'll see nothing, but occasionally you will see that PK and PD are being affected, and you know the neutralizing activity is actually having a clinical response. And same thing for any antibody. You can look at people that have positive antibodies and should look at whether the PK is affected by that. Are you changing the PK in those patients? Again, subset analysis, and this is actually routinely done by sponsors and agencies, actually. Now, the next slide, on slide 29, is simply a bioassay for neutralizing. I just show a cartoon of it to make it easy to see, where you have the product binding to the receptor and activating it. When you have neutralizing antibodies, you prevent that. And it just has to work out an assay that actually is responsive and sensitive to that. And that is something of an issue, as depicted on slide 30, because their serum itself can inhibit some of these activities. So you really have to be careful and do the proper exposure, do serum pre-exposures, post-exposure. And here you can see that there's a shift in the dilution curve where you're beyond what the serum is inhibiting to a point where you can see actually what the serum post-exposure is, and you're seeing, in this case, a neutralizing effect. So, in concluding, there's several take-home messages, I guess, that we'd like to make, and that is that the first one is that the results of immunogenicity testing rely wholly on the design and execution of the assays used to detect and characterize immune response. And because of that, it is impossible to compare rates of immunogenicity between biotechnology products unless a head-to-head clinical immunogenicity study is carried out. And that's what we see from some of the regulatory authorities, that they recognize this and they ask for head-to-head studies. But I think the other issue is that even when you do those head-to-head studies, the rates of immunogenicity are dependent on the assays, and so that when you do this comparison, the rates that you obtain may not reflect what's seen in the package insert from the innovator, because you're going to come a different output. And in fact, because of the assays and our understanding of these immunological assays are getting better and better and more sensitive, it's likely that when these such head-to-head studies do occur, that we'll see higher rates of immunogenicity than some of the older products have, just because of the improvements in the assays and the way we look at those things. So with that, I'd like to conclude and thank the audience for your attention, although I'm assuming you're paying attention. I can't see anybody. So thank you very much.
Dr. Czerny, it was a very informative presentation, and I think we got lots of details regarding the immunogenicity, and also a couple of points for further consideration. So, for instance, Dr. Czerny mentioned at the beginning that there was quite a lot of experience with immunogenicity investigations, but there is very little of these data published. And I think this is also an important issue, whether that aspect could be improved, how more publications may actually come out, and would that remain forever something that is a kind of stigmatizing the product? So during the development, there is probably an intention not to publish before it becomes clear what the immunogenicity is due to, whether the preliminary assumptions are right or wrong, and it's really a very complex area. So it's probably something that requires further discussion. Another issue is related to the assay. The choice of assay, which Dr. Czerny nicely explained, is very important, and is also interpretation of the data. And the third point is risk-based approach that is quite interesting, and I think it will be good to hear from the audience what are the views or comments on the points that were presented in Dr. Czerny's talk. I'd just like to ask one question, if you could pass that to Dr. Czerny. Chiara, in the conclusion and take-home message, I think there is a bit of clarity that may be needed. This comparison of the rates of immunogenicity is actually quite important. And as we heard from Dr. Pini, that immunogenicity assessment needs to be included and integrated in the comparability study. When developers of biosimilars are choosing the reference product, they are obviously looking at the immunogenicity profile as well. So this comparison of the rates is quite critical for the interpretation of the data. And I think when we talk about head-to-head comparison, it is possible to compare. The question is how to interpret the data. So for instance, if the originator had quite high rates using one assay, and then the similar product in comparing with a reference product using another assay, if these rates are higher or lower, I think there is a need for very careful interpretation. So maybe if Dr. Czerny could comment and maybe give an example on that aspect, just to clarify that it should be possible to interpret these rates, but it's not always straightforward, and it could be quite a challenge for both regulators and product developers.
Okay, so I had a question, I guess, about the rates and how do you interpret that. And I think the rates are one thing, but you know, as I said before during the talk, that the rates may have no clinical significance. So you always have to look at what the clinical significance is of those rates. The rates themselves, they are not that critical, that they don't correlate necessarily to clinical results. So you always have to look at what if you see a high incidence, what does that mean to the product? What does that do to clinical effect? Do you see neutralizing antibodies? Do you see that the PK is affected? Do you see safety events that occur? Anaphylaxis, infusion reactions. So it's really what's the output? What's the clinical effect? If there's no clinical effect, then there's really no issue about the rates themselves. The other thing is that you also have to look, because you can see neutralizing antibodies appear and disappear, that they're not long. So you have to do these studies for a period of time too. You just can't just say, oh, this has a neutralizing antibody, it's different. And of course, you got to look at the consequences of neutralizing or binding antibodies. Are they really affecting the PK? Are they affecting biodistribution? Do they affect the efficacy of the product in these in the clinical studies when you do these subset analysis? So I hope that answers part of the question.
Very good. Thank you very much. Now I see someone who obviously has energy in the early morning hours. So, Dr. Thomas Streitmuller.
Well, actually, it's not necessarily a question, rather than a comment I wanted to make. And the point raised by Barry goes perfectly in the same direction concerning the rates. The rates itself are not that important. At the very end, what really counts is the clinical impact. But what that means on the other end, and this is what we were discussing already yesterday, is really the importance of choosing the most sensitive population when studying immunogenicity, in order to be able to extrapolate these data to other indications. Because here you will not do a clinical study to investigate safety and efficacy, okay? But immunogenicity may have an impact there. This is, again, just to emphasize around this concept of sensitive population. The other remark, just very brief, to your request to publish more on these results, what actually will be the background for immunogenicity. I think that this is quite a difficult request. Difficult in that sense that you may only be able to speculate what can be a root cause for something, you know? Because in order to confirm that a certain attribute in a product really was responsible for immunogenicity, you would have to do a clinical study, actually. And would you really want to put the patients on risk, you know, in bringing into a population a product with a lot of aggregates and so on to see whether in fact the aggregates have been responsible for an immune response in patients? I really would question, you know, the ethics behind that. And this is why, in fact, it's actually quite difficult to publish these kind of results. This was only a remark.
Very good points. And yes, of course, we all agree that it's difficult to publish, but unless we stimulate the flow of knowledge, we'll actually never learn about that. So at least once the investigations are completed, I think it's worth considering publications, especially in some cases it may be more straightforward than in the others. We saw that for some products that were actually developed 20 years ago, the problem with impurities and the improvement of manufacturing process actually had a very straightforward impact in improving the immunogenicity profile. So there are certain cases that can be published. I just don't want to have a kind of excuse for the entire scientific community that, of course. But obviously, during the development of the product, when many things are not known, it is very difficult to get things out to the public domain. And also, this may also have a consequence of raising uncertainties and assumptions that the product may not be safe. So these are just points for further consideration. Now I see another person with plenty of energy. Inger, go ahead.
Discussion. Just a comment, because I think where we are today with the requirements for transparency and publication of data from clinical trials, I mean, the information will be published and it doesn't take long these days. So I agree there can be a challenge, a need for the older products, but I think with all trials running now, this is in good hands.
Are there other questions, especially from industry? What about Russian manufacturers? No questions to Dr. Czerny? Yes, there is one. Excellent.
Действительно, изучение иммуногенности является
Indeed, evaluation of immunogenicity is very important for all
но стоит отметить, что
biologics.
группа биологических препаратов тоже
But
является
nevertheless, we know that this group of drugs is
препаратов,
really very
базирующихся
heterogeneous.
на моноклональных антителах, тем
Some
более
of them
на
are really based on
антителах, которые часто
monoclonal
являются
antibodies.
комбинированы с какими-то продуктами,
Moreover,
с малыми
new
молекулами.
antibodies
Здесь возможность
are conjugated
иммунного
with
ответа
some
на
other
чужеродный
products, such as
велика и
small
необходимо здесь
molecules.
оценивать. Что вы думаете относительно
And these
препаратов, которые не являются
Types of
для
molecules are really
человеческого
highly
организма природными
immunogenic,
антигенами.
so it should
Природными
be
гормонами, например,
evaluated always.
другие молекулы. Насколько здесь необходимо
But what do you think
проведение
about the drugs
долгих сравнительных
that
исследований
are not really foreign for the human
также
body,
необходимо.
such as natural
Учитывая то,
hormones,
что в норме в
insulins,
организме
other types of
существуют
natural
все эти
molecules?
антитела, идиотипические, антиидиотипические антитела, которые не всегда оказывают какое-то вредное воздействие именно на молекулы,
Whether in
которые
such
являются
cases
для
we really
организма
need to
более
conduct
чужими.
long
If I understand the question, many of the proteins that we have are endogenously produced. Insulin is one of them, human growth hormone. There's many of the products that we do have, and therapeutic proteins are in fact produced in the body as well. And so these are in particular cases where you really want to make sure that you're not neutralizing the endogenous activity. There's obviously been cases of pure red cell aplasia or thrombocytopenia where you have neutralized the endogenous activity of the enzyme. So you want to make sure that the product that you're using won't do that. And so these assays, I think, have become very important to have neutralizing assays, particularly when you have a non-redundant function for these, which TPO and many of these products are, is that you don't want to be causing a clinical sequelae that are critical to the patients. So I think in these cases, it's imperative that you do these neutralizing antibodies for endogenous proteins that you might be impacting.
Because I do want to make one comment on this question related to the need for looking into antibody formation for insulin. The example I mentioned yesterday where we did a quite drastic change in an insulin process, it was absolutely required that we also ensured that immunogenicity as a critical attribute, that's also comparable. So I think you need to see this sort of in the overall perspective of the comparability exercise, consisting of both the quality and the non-clinical and the clinical part. So if you see a significant difference in the immune response for two insulins, you would immediately start wondering what's wrong here, what has changed, and want to get that into control. And that is regardless, also, although so far, and I agree to that, is not seen negative clinical impacts of the antibody response for insulins.
Thank you, Inger. Last comment to
If I may
Alex.
just say that there is a very good example of relevance of the data on immunogenicity with endogenous hormones coming from the history of follicular-stimulating hormones, FSH. If you recall that 20 years ago we had urine-derived products which were available from different sources, and they actually accompanied with numerically higher levels of immunogenicity. That prompted development of recombinant nature of FSH, which actually were dramatically better in terms of immunogenicity and safety. And now we're coming into era of biosimilars of FSH, which actually have even lower immunogenicity potentially. So that's just example how endogenous products or hormonal nature products may have immunogenicity, and how the manufacturing and evolution of the process of development actually resulted in improvement of immunogenicity.
excellent reminder on the history. Thank you very much, Alex. So we'll move now on with the other presentations.
Distinguished colleagues, I thank you for the opportunity to address the participants of this conference. Practically all previous speakers, colleagues participating in this conference dwelled on the definition of the biological product, biological substance, biological similar, biological analogs, the main causes of the heterogeneity of protein molecules, potential differences in their pharmacological properties. So let me go straight to some practical issues dealing with our routine everyday work. All of you are aware and it was mentioned on several occasions today and yesterday which particular drugs are attributed to this group and how they are classified. And I would like to go in some details in the part of classification that following the decision of coordination group on the procedure of mutual recognition and decentralized procedure on medicinal drugs, also attributed to biological drugs, the low molecular heparins and pancreatins, which are regulated starting from 2006 by the law, and in 2007 the list was expanded in Russia, which is of particular importance because here in the country we have on the market a huge number of such drugs obtained from the organs and tissues of humans and animals, such as bacterial lysates, etc., etc. So this is not at all an inclusive list presented on this slide. All these products actually belong to biological products, and all of them, as it was already mentioned by numerous speakers, including Carlo Pini, all of them should undergo complex tests and studies recommended by directives European Medical Agency, studies covering the biological drugs, and they cannot be registered following just the results of routine tests in the frame of pharmaceutical expertise. Some of them can be treated with the concept of biosimilarity. As for biosimilars, in Russia there is a legal definition of this group of drugs, and as for concept on studies of these drugs and expertise of the results, this concept is still under development. In our center, FSBI of expertise of medicinal products or medical products, we summarized international expertise and definition of biosimilars, biosimilarity. We use at our center the latest review, amendment, and there's also our proposal to amend the Federal Law 61 to amend with and there is a definition of a biological drug given in that law. In our everyday work, to harmonize the expert requirements with the European community, which is the major vector of our activity in perfection of our performance, and with the expertise of evaluation of originator and generic drugs, we take into account international experience, including European Medical Agency experience in particular, prepared guidelines developed by our center are harmonized with the European approaches and also accumulated regulatory experience of the Ministry of Public Health. As for regulations stipulated in Act 61, I would like to focus on Article 14 that pertains the use of medicinal drugs. The use of medicinal drugs existing in our market for more than 20 years. Thus, these drugs should not be subjected to clinical studies. This approach, this norm, is of particular importance because many biosimilars, because this can cover multiple biosimilars theoretically, and these drugs can be registered without or marketed without additional clinical studies. Although biosimilars cannot be an exact copy of the reference drug, that is why experts keep to the opinion that application of this law, the use of this law is the whole group of biological drugs is not possible, and this issue can be solved in the nearest future due to amendment of existing law, and the Ministry of Health raised this issue on many occasions. A good example, if you don't mind, example in the previous topic, one of the oldest representative of low-weight heparins, enoxaparin, which was marketed in Europe in 91, in Russia in 92. This enoxaparin can theoretically be treated as a drug registered and existing on our markets for more than 20 years. But because of the origination of active substance, biological origin, and impossibility to provide safety by routine studies, European agency recommends to apply the concept of biosimilarity to enoxaparin. This position is shared by our center and leading experts, including Society of Cardiologists and clinicians in particular, think that analogs of low molecular heparins can be marketed only following the provision of the results of clinical studies, comparative studies of efficacy and safety with the reference drug, and also studies of bioequivalence of the biosimilar with the originator. I would like to underscore the word bioequivalence with respect to any medicinal drugs that is viewed as a biosimilar or as a generic. You should know they should fall into a specific range. A specific parameter such as immunogenicity, for example, cannot be worse or better. They must be equivalent to reference drug for qualitative interpretation. Until these corrections or amendments into legislation, the clinicians, before the corrections are made, the clinicians think that low molecular heparins can be used in a particular patient only after there is a verification of the pharmaceutical and pharmacological, but clinical equivalence as well. So they do not accept interchangeability of low molecular heparins. Let's speak a little bit about development and planning of the scope of studies and expertise of the results of those studies. Specific attention during the planning stage should be given to the correct choice of a comparison drug. Studies should always be comparative. Studies should use some reliable tests, orthogonality principle based on different principles to evaluate the same parameter. You should always keep to all specific stages in the development of the drug, specific attention to the quality. After that, preclinical studies, and only after that, clinical phase. The diagram that you see on the flight, you should pay attention to the scope of each stage and financial issues. As you can see, verification of physical, chemical characteristics and quality at the stage of organization of manufacturing, at the stages of quality control, at the stages of preclinical studies. So these costs, financial, intellectual, much higher than costs of clinical studies per se. And the scope of each stage would depend on the results of the previous stage. And also you have to take into account the fact that when you get results proving differences between the originator and reference drug and biosimilar, such difference bring you back to the previous stage, almost down to the manufacturing process. And if there are clinical significant differences, we should review the manufacturing process either, or recognize that we speak about biodifference, not a biosimilar. Thus, a new product should be studied using the program, comprehensive program of studies of a new product. Let me summarize dates mentioned by previous speakers on specific stages of the development and type of studies that are used. This schematic diagram shows general principles that you should into account at the stage of planning, the introduction of a new drug into clinical practice, from the manufacturing process to marketing and pharmacovigilance stage. So we should follow the green line from one stage to another. Otherwise, if there is a failure, not a success, you should go back to the previous stage. Again, the most expensive are the organization and validation manufacturing process and quality control of the final product. And if you have, and sometimes you have to return to the manufacturing phase, even following the results of the preclinical studies. If you, for example, get differences in pharmacokinetic parameters, you have to go back to the manufacturing stage and start the whole process anew. So let's analyze the first stage. This is the quality control. Specific attention, and so it is in our everyday work, and it also concerns the manufacturers of biological similar. Specific attention is given to quality. This is pharmaceutical quality, non-comparative parameters characterizing the product, such routine as description, sterility, pH. Standards, they're all standard requirements to any medicinal drug. But the main thing is a comparison study with the reference drug for verification of similarity, following such parameters: authenticity, impurities, biological activity, stability, bulks of pharmaceutical substance, and final medicinal product. And focus your attention on the following: that the results of the study's biological activity can be used later on in the future to confirm the special activity in the frames of clinical studies. To demonstrate, I give you another example. This is the development program of one of the oldest biosimilars from the group of monoclonal antibodies, rituximab. As you can see from these slides, authenticity was studied by numerous modern reliable methods, and similar orthogonal methods were used to examine impurities, non-active substances. Some few words later about biological activity. This slide shows perfectly the scope of studies of the original drug, innovator drug, and the scope should be used by all manufacturers of biosimilars. Of course, the list of procedures is not a must. It can be changed following modern achievements, scientific progress, but as for today, following the result of our analysis, you can see this specific list of studies. Thus, recommendations of our center, FSBI, recommendation on compiling the registration dossier. You should take into account, while preparing the dossier, the requirements of European guidelines, specification, analytical methods, acceptability criteria of biotechnological and biological drugs. These recommendations are announced by the director of FSBI, Mironov Alexander Nikolaevich. You should pay specific attention on specifications on the quality control, normative documents. It's not a document for description of physical, chemical, pharmaceutical, and biological properties, but nevertheless, normative document covers or includes some methods that are not justified for the control of a particular product. The number and types also do not correspond to European recommendations or guidelines. The quality of the dossier, unfortunately, quite often absent or limited very principal data, development, statistical data, detailed description of the manufacturing process, validation of the process, control of quality, analytical methods, and validation of analytical methods. So my request to describe all those procedures and their results and to discuss them in your files or dossier. Quite widely used in the practice when you declare specific parameters, sometimes antiviral or viral safety or presence or absence of specific impurities. So the manufacturer guarantees the absence of such impurities, but there are no documented reports. Nevertheless, next stage is preclinical studies. Main principles. The scope of the study depends on the degree of biosimilarity confirmed at the previous stage of quality control. It should include verification of the comparison-specific activity in vitro and in vivo on relevant models, reliable, validated models, comparative toxicity studies, minimum 4-week subchronic toxicity studies, also local irritability, toxicokinetic analysis should be included, as well as immunogenicity studies results. In some cases, when there is a scientific rationale, pharmacodynamic and toxicity in vivo studies are not a must because they're not always sensitive enough to detect potential differences. But all those should be discussed and it should be proved or justified. These principles can be applied to all groups of bioanalogues. Another early promised clinical example where specific activity is studied. Here on the slide you see a program of pharmacodynamics evaluation, pharmacodynamics in vitro evaluation of infliximab. As you can see, binding with antigen, target with specific receptor, cross-over activity with different human tissues. All these studies were very extensive, requiring a lot of time, intellectual efforts, money, scientific experts. And again, I tell you that it's not a limited, comprehensive program for infliximab only, but just scientifically justified example. We are open to other modern scientific approaches that can be used in such development programs. Now, recommendations of FSBI SCEMP on registration dossier, and particularly in the reporting the results of preclinical studies. It is recommended pay attention on justification on the development of preclinical program, execution of reports. Often gives no chance to determine their authenticity. There should be detailed description, method, results, statistical treatment and analysis. Not only resume and some recalls in the investigators' brochures, recalls of the results of some performed studies. So we need all initial data, primary data. Next stage is clinical development of a new drug. Common or general principles. The scope of the study will develop on the degreeification of biosimilarity at the stage of quality control and within the frames of preclinical study of biosimilars, and pharmacokinetic and pharmacodynamic properties during the first phase are a must, strictly obligatory. During phase 3 studies, one should examine endpoints sensitive in terms of difference to the comparator drug or reference drug. And of course, these endpoints may not coincide with the endpoints studied with the original or reference drug. A safety database, including immunogenicity, should be accumulated for at least 12 months after the completion of clinical trials. All this is applicable to all groups of biosimilars. Another clinical example to demonstrate all these stages, clinical development of filgrastim. As you can see on this slide, we studied pharmacokinetics. It was a crossover study with single subcutaneous and intravenous administration of the drug. Primary endpoint is AUC, area under the pharmacokinetic curve. Secondary maximal concentration and a half-life time. Pharmacodynamics was studied on healthy volunteers. Different doses were used. Primary endpoint is the absolute white blood cells count, secondary number of CD34 cells. Efficacy was studied with prophylaxis of severe neutropenia after cytotoxic chemotherapy in a homogeneous group of patients. Primary endpoint is duration of severe neutropenia. Absolute count of neutrophils should be lower than the value presented on this slide. Secondary frequency of febrile occurrence of febrile neutropenia, infections, and cumulative dose of granulocyte colony stimulating factor. Safety. Time of follow-up should not be less than 6 months. Immunogenicity. Time of duration of data collection should be no less than 12 months after completion of the study. Recommendations of FSBI recommendations on compiling the registration dossier. When you prepare the results of clinical studies, it is recommended to provide scientific justification of clinical development program with due regard of the results of comparative studies, quality and preclinical studies. There should be justification of the examined surrogates and clinical endpoints. The duration of safety studies in your protocols is not sufficient in majority of cases, so you should give enough attention to this issue, and there is no description of pharmacovigilance measures. You should always envisage them. Pharmacovigilance of clinical safety should be continuous with a regular evaluation of benefit-to-risk ratio. You should always take into account requirements to safety monitoring, requirements that are applied to the comparator drugs, comparison drug onto the whole group of biological products. General recommendations of FSBI on compiling the registration dossier. This is the conclusion of the development program for biosimilars. While preparing document for marketing biological products or biosimilars, bioanalogues in the Russian Federation, you should take into account European guidelines as well to provide the results of the studies with detailed initial primary data and the analysis, validation, statistical treatment of the results, not only reviews or resume. Your studies, you shouldn't forget the necessity of comparative studies when you change the formula, composition, place, manufacturing place, etc., etc. You should provide the volume of documents, documentation that meets European requirements that you usually fail to provide. Thus you lose time, etc. Immediate and delayed consequences. You should thoroughly prepare all these documents, signatures, stamps, etc., so that we never doubt their authenticity. To conclude, interim results of harmonization, the international requirements, ICH and the EMA, we following foreign and our own experience on FSBI, we can apply to biosimilars all chapters, non-clinical studies of safety with the purpose of conducting clinical trials, medicinal products, studying pharmacological safety of the product, general principle of conducting clinical trials, drawing up a protocol for a controlled clinical trial of medical products, statistical principles of conducting clinical trials, structure blockchain content of clinical trial reports, bioequivalence studies of generic drugs, non-clinical assessment of safety of safety of drugs produced by biotechnological method, quality control, non-clinical and clinical studies of biosimilars, and assessment of immunogenicity of therapeutic proteins produced by biotechnological methods. Possible, I'm looking for your comments, your proposals for further review and amendment. We will be really thankful for your point of view, key directions and questions or issues for further development. We pay specific attention to further development. We actively participate in preparation of changes into law enforcement. So we call on all participants who develop biosimilars, their attention to quality control, preclinical clinical studies, training of all participants, pharmaceutical distribution, scientific guidelines on separate groups of biosimilars. They were announced by the director yesterday. And to conclude, I want to tell that without introducing GMP, GLP, GCP principles and standards, as well as pharmacovigilance and marketing and other principles, you will always fail to provide quality, safety, and efficacy of biosimilars. Thank you for your attention.
It's a great honor for me to be present here and to speak in front of you and in this very representative forum. I want to discuss an issue suggested by the organizers: clinical application of immunogenicity of biological drugs, medicinal drugs. You heard a lot already about biological drugs per se, and about aspects of clinical impacts of immunogenicity, impacts as related to safety and efficacy of drugs. So, if I repeat something, I apologize beforehand. This is because the issue is specifically important for further discussion and for further actions as well. All of us speaking today at this conference start with the definition of a biological drug, and each speaker underlines that it's a principally new medicinal drug different from whatever available in the group of low molecular drugs, and differs by so many aspects, features. By molecular weight, by complexity of structure, by specific attributes, quality, stability. But we do realize that these are drugs of biological origin. In most cases, they are proteins, and we, based on even our high school knowledge, we do realize that foreign protein is always an antigen, a real antigen. So, the main associated implication property is immunogenicity. So many factors interfere in modifying immunogenicity, maybe enhancing. This is synthesis of antibodies, first of all. This is induced synthesis of antibodies. This is the structure of the molecule per se, attachment of different additional groups which change activity of their molecules. There are particular technological factors that also induce synthesis or facilitate synthesis of antibodies, such as impurities, additional isoforms, or what else important for immunogenicity. It is formation of aggregates during the manufacturing phase, manufacturing of the final formulation. It was already mentioned that induction of antibody synthesis observed in cases of subcutaneous administration, in case of chronic administration, also in administration to specific groups of patients or populations of patients. But we should admit, dear colleagues, we still don't know to the utmost why part of biological drugs are immunogenic and induce formation of antibodies, and the other part does not induce formation of antibodies. Thus, mechanisms of immunogenicity development are not still clear to all of us. Following the package leaflets attached, we can always judge. We can find in those leaflets the information of our formation of antibodies, the rates of formation of antibodies. So, if we just read these leaflets on monoclonal antibodies, then you'll see immediately that incidence or rates of formation of antibodies in different groups of patients with the use of different types or groups of monoclonal antibodies, these incidents are various. And you see decreased synthesis of antibodies when we move from chimeric or recombinant antibodies to humanized, fully humanized antibodies. Besides, very important is a particular population where you administer drug and study the rates of immune reaction. You see on this slide that such incidence of immune reactions is much lower in patients getting methotrexate. But because this is a drug that inhibits induction of antibody synthesis and formation of antibodies, this is we observe a few-fold lower incidence of immune reactions in such patients. You see similar information on formation of antibodies to interferon beta. You see multiple studies performed. They were long-term studies. The result that you see demonstrates that the incidence or rates of formation of antibodies also varies widely depending on patients' population participating in the study. Depends on the dosage, on the therapeutic regimens. You see 2 to 22% of patients with antibodies to interferons. If we summarize all this info about the incidence of immune reactions in patients to different biological drugs, then you'll see that practically all biological drugs can induce the formation of antibodies, so the problem exists. So that's a challenge. So, this issue should be discussed and should be regulated by the law. But my particular objective is to discuss clinical implications when we have such cases of antibody formation, and those clinical implications result in loss of efficacy of biological drugs and result in crossover reactions with endogenous molecules present in the human body. And we also observe hypersensitivity reactions to administration or introduction of foreign proteins. We usually observe anaphylactic reactions or serum reactions as demonstration of hypersensitivity or manifestation of hypersensitivity. We are the clinical signs. What are the clinical signs that help you to identify formation of antibodies? You see multiple studies presented on this slide. One of them is evaluation of the impact of neutralizing antibodies to beta interferon on the course of systemic sclerosis. So, these studies demonstrate that treatment with interferon beta, with simultaneous emergence of antibodies, of course, reduces the efficacy of treatment. And here you see the lists of some of the studies that speak actually or state emergence of such an issue, such a problem. Another important clinical study which analyzes efficacy of monoclonal antibodies in patients with rheumatoid arthritis. And here you evaluate the efficacy depending on the induction and the incidence of immune reactions in patients. So, you observe patients whether with a stabilized disease or remission, or you have a result the progression of rheumatoid arthritis. Here you see the first diagram in patients with no neutralizing antibodies. The efficacy of their drug is much higher as compared to the group of patients where we observed the induction of antibody formation. You see the diagram in the middle demonstrates you that the efficacy gets lower with the increase of antibody titer. If we have less than 100 units per milliliter, we can achieve some stability or stabilization of rheumatoid arthritis as evaluated by DAS28 less than 3.2. If the titer of antibodies is greater than 100 units per milliliter, you practically don't see patients who manage to achieve stabilization of their disease. So you see similar data with the Fabry disease again. The higher is the titer of antibodies to galactosidase, the lower is the efficacy of treatment. And it is really so that a specific issue with all those cases is evaluation of immunogenicity in emerging biological drugs, because these, or generics, because they are trying to be as close as possible to the originator drug. But nevertheless, there are differences in the manufacturing procedure. Impurities, aggregates, and raw material that is used. Thus, what is differences results in different immunogenicity properties of generic drugs as compared to originator or their reference drug. So you already mentions the necessity to develop specific regulations, guidelines, which would regulate the method used. Duration of studies, specific tests. Because it is a real practice when a manufacturer of biosimilars would try to use different evaluation methods and analytical methods, and would provide thus different results of those tests' results that would differ from the test of the original reference drugs. So, it's a special issue when we speak about immunogenicity. Special issue is the evaluation of immunogenicity of emerging biosimilars. generic drugs, as these drugs are trying to be very similar with the original biological drug. But nevertheless, they differ with the initial raw material, with the manufacturing procedure, and there are differences in impurities and aggregates. So, the conclusion is that those biosimilars or generics that emerge in the market would differ from the originator or reference drugs by immunogenicity, first of all, immunogenic properties. It was already discussed. The necessity of the development of specific documents, guidelines, recommendations that would regulate evaluation of immunogenicity, used methods, time frames, populations, patients' populations. Because it is really so that the manufacturer of biosimilars can use or would use different evaluation methods, analytical methods, and would finally give the info result. In info in a different way as compared by the manufacturer of the original product.
Проблема взаимозаменяемости этих препаратов должна обсуждаться, и должны быть предприняты какие-то шаги, чтобы эта замена не производилась бездумно только потому, что
we need to standardize all those regulatory documents that are used when we register biosimilars or generics or biological products. Why it is important? Gerapharm company. My question to Professor Zырянов. Could you tell us, please, whether we should really be alert with the issue that the use of biosimilars developed not in compliance with existing norms, that such use can induce cross-immune responses or resistance? Are there any clinical cases reported so far? Or it's just a theoretical assumption or speculation? Thank you for your question. If you are involved in such issues, you should be aware that biosimilars that came onto the market at the end of the 90s or beginning of 2000s were causing such effects, sequelae, due to their immunogenicity. We know multiple such cases. So, it's not just a theoretical assumption. There are cases with immunogenicity of erythropoietin due to changes in the manufacturing procedure. So, it's one of the major issues that should be studied from my point of view, and I'm a clinician, within the frames of clinical studies. As a clinical pharmacologist, I trust only the results of clinical studies.
Okay, please. Thomas Kirchlechner from Bandos. I have a question to the last speaker, to Professor Zyryanov. Sorry, my pronunciation. Professor, you talked about biosimilar erythropoietins that have more aggregates or more isoforms and caused clinical effects. I would like to remind you to make a clear distinction between high-quality biosimilars that are approved by European regulators, which have even been recognized by regulators to sometimes be even more pure, have a higher purity and lower levels of aggregates than the original product due to the more modern technology, in sharp contrast to other products that may be around in different markets of low quality that are not compared properly to a reference product and would never be approved by a stringent authority like the EMA. Those products do not deserve to be called biosimilars. They are non-comparable biologics, or I would even call them bio-questionables, if you allow this term. Thank you.
Well, indeed.
Well, indeed, when I was talking about biosimilars that I discussed and when I referred to the professor's study, if you remember, they were taken from different countries of Latin America, Southeast Asia, from countries where there are no stringent requirements to the studies of biosimilars. So this explains, from my point of view, the lack of stringent requirements. This explains emergence of such drugs with very low quality properties that were not properly studied. Does this allow them to stay within the market? So, more stringent requirements will bring the number of such low-quality drugs to zero, hopefully. Thank you. Ravin Yazov, you commented the study of 2007 and you demonstrated there were biosimilars with different pH following the requirements of European pharmacopoeia. We cannot call those drugs biosimilars. It's just lack of quality, pharmacological. And these are issues of GMP, not similarity. And then again, you commented high levels of aggregates, which means that biosimilarity was not really proved. So, these drugs should not be viewed as biosimilars. They are just bioquestionables. When we speak about biosimilars, it means that they are very close to original drugs. They do not differ practically from the immunogenicity point of view from the innovator products, from reference drugs. Well, I want to comment that those drugs that I demonstrated to you, they were also attributed or given INNs. They were circulating and they were procured in the market instead of original drugs. In real life, they were viewed as biosimilars and were used for treatment of patients instead of original drugs.
Inger Mollup, Novo Nordisk. I think the last two talks raise a very interesting and tough question. Talking about mentioning, as Dr. Zirinov listed out the products that have been approved to a less stringent standard and that are still on the market. I think I heard mentioning that there'll be post-marketing follow-up, but I would like to ask sort of the question to the last speakers. So will there be other ways of ensuring that products converge to the standard that Russia is deciding on? So any other regulatory actions that would be interesting to know?
Если позволите, я отвечу.
I would get back again to the results of enoxaparin studies by Professor Zyanov. There is another important issue continuously ignored by mass media and scientists. We should keep in mind that ensuring compliance of a particular drug and medical drug on safety, efficacy, quality, and other parameters to an original one, and this involves not only biosimilars. It's not just the issue of marketing authorization. None of the applicants would not demonstrate low-quality product. So, those biosimilars that were commented, they were taken from the market, from the circulation, and the differences that were observed in their properties, pharmacological properties, and routine quality parameters demonstrate that manufacturer that not comply with the procedures, manufacturing procedures that actually we observed during pre-registration studies. So, stringent requirements during registration process and quality control of preclinical and clinical studies are of importance. Pharmacovigilance should also be in place. You should realize that with issuing of marketing authorization, the questions are not answered. Here, pharmacovigilance should be in place. Authorities dealing with marketing authorization cannot help with withdrawal of low-quality drugs from the market. It's the mission of pharmacovigilance.
Мне кажется, данная дискуссия возвращает нас в начало конференции.
I think this discussion brings us back to the very beginning of the conference and your presentation where you spoke about the risk assessment. So you mentioned that as for today, there are registered biological drugs, registered at different times and with different requirements circulating in the market. So, regulators should make a decision based on the experience gained during registering of these drugs. What should be done with biological products registered against a different set of requirements? So, my question is whether the WHO and their experts plan to put together to develop a document which could provide a guidance in this particular direction.
Lena, this is a very good point, and I also would like to mention that the entire session was really fruitful and a lot of important points were raised. So half of the points are very specific and related to the immunogenicity, but the other points are also related to some general aspects of naming biosimilars, as we discussed yesterday, stringent versus flexible regulation and these type of issues. So I think that it's really setting the scene for further discussion among all relevant authorities in the country, and it is indeed important to follow on that and to have regulators and manufacturers initiating that discussion because it will be of critical importance. With respect to the specific issue of regulatory action, I think we made it clear yesterday that the role of regulators is extremely important, that they need support from the Ministry of Health in doing their job, and specific action in terms of the products that were licensed with probably less data than it would be required today during the time when requirements were not defined is indeed the next step to be taken. So we do plan to continue development of the guidelines. The options that I presented yesterday will be developed further and will also go through a very detailed discussion and consultation with all regions. And I think with respect to this audience, it will be very important to establish some sort of network for future communication so that we have feedback at the next stage of the document development and also examples from this area of the world, which is actually important. So the actions that we are talking about while leaving product on the market but requesting additional data will obviously have certain implications for manufacturers and will be a question, would it be feasible in all cases or not? So we plan to develop also some case studies to see what is the feasibility of each approach and how to balance regulatory action with the actual response from manufacturers in the interest of patients and in the interest of access to these products. So there will be definitely another year or two of consultation that will take place. And we hope that we'll establish proper communication channel so that we can actually get your input into this consultation. Thank you.
I always start all my talks with this, because this is why our program is there, pharmacovigilance. I also show a color photograph of this event, which was already reported in black and white in the 60s, but this is a picture that's much more recent. So as long as we'll have medicines that can do this, we can never take it easy. We'll always need to have pharmacovigilance in place. So, but based on that event from the 60s, a resolution was created. As you know, the World Health Assembly is a forum where countries from the world meet to discuss problems of concern, problems that are affecting their populace. And here, in 1963, at this assembly, a resolution was raised that invited member states, because of the event due to thalidomide, to arrange for a systematic collection of information on serious adverse drug reactions observed during the development of a drug, and in particular, after its release for general use. And of course, this was in the 60s, and since then, we've come some distance. We do have such a program in place now. We do have a network of 146 countries that have pharmacovigilance systems in place, but that still leaves about 50 countries that do not have it yet. And those countries that do have pharmacovigilance systems are not entirely up to speed either. So just having a pharmacovigilance center in place does not necessarily mean that the capacity exists. So, of course, the first step is to create those centers. So, just to give you a very broad overview of the WHO program for international drug monitoring, which is the field name for the WHO pharmacovigilance program. It has the WHO headquarters in Geneva. And then we have, many of you know, the Uppsala Monitoring Center, which is our lead collaborating center for the work in medicine safety in pharmacovigilance. It is based in Uppsala. So headquarters takes care of policies and strategic development of the program, and Uppsala traditionally has been the center for collecting the data coming from the use of medicines worldwide into one central database on behalf of WHO. And then more recently, in 2009 and 2010, we've had two more centers joining the program. One is in Accra, in Ghana, in Africa, and one is in Rabat, in Morocco, also in Africa, but in the WHO regional distinction, we call it the Eastern Mediterranean Regional Center. So we have two centers that have come up. The Accra Center takes care of pharmacovigilance capacity building in the sub-Saharan African continent, and the Moroccan center, the center in Rabat, takes care of capacity building for pharmacovigilance and technical advice for pharmacovigilance in the Arabic and in the Francophone world. And then we also have another collaborating center, not very well known, but still doing a very important piece of work, and this is our collaborating center in Oslo, in Norway. This center is responsible for developing the ATC-DDD. I would like to see, by a show of hands, how many people have heard of ATC-DDD? Okay, I see 1, 2, 3. Okay, still very few. And so only about 5 or 6. So that shows that it is not enough to do our work, but we also need to be visible and promoting our work. ATC stands for Anatomic, Therapeutic, and Chemical Classification of Medicines. And DDD is the defined daily dose. So you can have your medicines, INN, is accorded to it. And then, of course, you've got to classify, you've got to put it into little boxes. And that is done by the center. And not only do you need to do that, you've got to give them the defined daily dose, which will then allow us to understand the utilization pattern and consumption of these medicines across the world. But of all the centers, to my mind, the most important is what you see at the bottom of the screen, and that's the national centers. If we did not have the National Pharmacovigilance Center, we do not have the country represented in the program, and then we are irrelevant. So at headquarters, as I said, we are responsible for policies and strategic development of the program. We want to make sure that what we understand from one corner of the world is quickly shared with the rest of the world. And that is the exchange of information. We do it through rapid alerts, we do it through newsletters, we do it through our website, and we also do it through electronic platforms, such as the Vigimed. Some of you may be members of that. We develop policies, guidelines, and normative activities that, for instance, to decide that we need urgently a policy on biotherapeutics, on how we are going to develop risk management plans. What are the elements for the risk management plans in biotherapeutics? And that would be to have such a direction would be a policy statement, and then to develop the support structures and guidelines would be the next step. So these kind of work would be undertaken at the headquarters. Once we have these in, then to take them to the country, work with the countries for the adaptation of those guidelines would be the country support, and we collaborate with any program that uses medicines. And by medicines, of course, I mean a much broader understanding of it, medicinal products, including vaccines and biologicals. So any country that would use medicinal products, we make it our business to work with them. And any program that would use medicinal products, we make it our business to work with them. That's what I mean by collaborations. And all of this takes money and people. And so we also engage in quite a bit of resource mobilization. That is, we talk to Gates, we talk to Global Fund, we talk to UNITAID, and we make sure there is money for our program as well as money for the countries. It's not always easy, though, that bit. And of course, advising on all these steps and our understanding and improving our understanding and guiding on every step of our work is a big advisory committee of 12 members, chosen in their individual capacity as experts, but of course with the approval of the governments. And there are 12 members. We try to keep an equal gender balance in this of 6 men and women. And they are chosen from various fields of expertise in medicine safety. And they meet once a year to advise us and help us in the process. See, WHO pharmacovigilance strategy, what defines it? How do we do our work? How do we decide what needs to be done? That is, understanding what's, first we have to listen to the countries, understanding what's available and what's needed in countries. So often we would base this on bilateral discussions and also through multilateral assessments and analysis. And consistently, even though I showed you the previous slide, it was a 2008 assessment, but consistently we've seen that. Many assessments have followed that, and we've seen that lack of pharmacovigilance looms, continues to loom as a big problem in many of the low and middle income countries. So that will continue to be our challenge. And even when centers are there, they are not resourced well. There is usually one person dealing with pharmacovigilance for the entire country, and so on and so forth. This then decides how we have to position ourselves in supporting the countries. So that's how the strategic program decisions are made. Where should we put our efforts? Which way should we be moving? And the kind of assistance, technical support, equipment, training, money, and so on. So with that as our starting point, this is what we looked like when we started. And then, now, this is what we look like. So 10 years ago, is what I showed you, and this is what we look like currently. That is, all the colored spots in the map are those countries with pharmacovigilance systems in place. But the colors, the lighter blue on the map, are those countries that have a system in place but are not really functional, are not really up to speed. If I would sum up the WHO pharmacovigilance strategy, I would say it is a three-pronged approach. The first is to do what we've done for the last 40 years of the program. This is our as before strategy, building on the strength of the WHO program. And as many of you would know, our program of 40 plus years has been built on spontaneous reporting. That is, to the alert health professional will make an observation of, will have a suspicion of an adverse reaction in a patient and will forward that to the database, the national database, and eventually to the global database. So that is the spontaneous reporting system. And the database that I mentioned, which is maintained in Uppsala, is primarily a database of spontaneous reports. So that is, we will continue to do that because no matter what the limitations of this methodology, this is the bedrock of pharmacovigilance in being the easiest and the simplest to establish, and also in being able to detect rare events, and also being necessary when we are talking about long-term strategies. And then, of course, we are not saying this is all that we will do. We need to do more than what we've been doing, and that is to make current methodologies stronger, which would mean focusing on limitations of spontaneous reporting. All of you who are working with spontaneous reporting know that it is difficult to get numerator. How many will be affected, say, with lactic acidosis, with one of the HIV medicines? How many, that is, the quantitative aspects are difficult to discern because it's a very voluntary process. So therefore, not all the reports that are occurring are ever making it to the database. And nor will we be able to ascertain conclusively what is the denominator, how many people have been exposed to these medicines, because unlike the advanced settings where you do have these kind of data available to you readily, in low and middle income countries, you do not have utilization data. So the denominator is not available. The numerator is very subjective to individual reporting. Therefore, your rates and frequencies are difficult to get. So therefore, clearly, we need other methods that will complement the spontaneous reporting system. That will, because the spontaneous reporting system, we will say, is a hypothesis generation tool, then we need other methods to substantiate what we've seen. And therefore, we would need, for instance, active surveillance methods, registers, and so on and so forth. And then we will like to do things that we haven't dared to touch so far. For instance, we haven't really brought patients on board effectively. So the last couple of years, we have been brave enough to work with patient organizations, to work with consumer organizations, to understand how they could support the pharmacovigilance process. And we have developed guidelines on how pharmacovigilance centers worldwide could prepare to receive reports from patients. And we have also developed tools that patients could use when reporting to the pharmacovigilance centers. And of course, in there, you will see, in the never before, you will see, I have also put in biotherapeutics. That is something we are very scared, but we cannot look away. So you have to teach us on how we should be moving forward, and you have to help us develop good guidelines, good practices, and good methodologies that we can put in place with your help. So that is something that I have put squarely in the as never before strategy. And then monitoring and assessments. We can have all these wonderful things in place, but if it doesn't deliver, it's not even there, in my opinion. So just to make sure that what we develop will lead to something, we would like to assess the structures, the processes, and what do they lead to, the impact ultimately, using usually surrogate measures in pharmacovigilance. Okay, I'll bring us to the concept of generics. In all public health programs, where we are attempting to tame the beast of many millions of people requiring medicines, the concept of generics has revolutionized the way we make medicines available to patients. Because when the patent expiry of medicines, with the patent expiry of medicines, we can move into generics, which can make the medicines available to many more patients due to the overall cost savings. And why are there cost savings? Because generally, in generics, we can use data from investigations with innovative products. That has been the broad theme with so-called traditional chemical generics. This, of course, as I said, has revolutionized the way we have made medicines available to patients worldwide, in HIV, for example. All the parts covered with red have been able to get access to HIV medicines because of the concept of generics. But as I said, this is easier with traditional chemical medicines because they are relatively small, stable molecules, and no need to repeat large-scale human trials. We've been hearing this the last several lectures now. But safety monitoring is needed nevertheless. I'm not saying that generic medicines we can throw away the key, just put them into the countries and walk away. It's not the case. So we will continue to battle that pharmacovigilance needs to be in place with also for generic so-called chemical medicines, because they would be used in other environments and populations in the low and middle income countries with their specific comorbidities. Onchocerciasis, how many has heard about it? But it will be there in these settings. And these medicines will be used alongside treatment of onchocerciasis, alongside treatment of MDR-TB, alongside treatment of river blindness, and so on and so forth. So we have to be constantly monitoring these medicines because of the genetic differences, because of the different use environment, and of course, because of the long-term effects. So I'm going to skip this, but just to highlight the point that this has been a battle for us in WHO, that the procurement and access programs have looked at medicines as the solution. Medicines are the solution if we will use them, if we will consider all the underlying factors when using them. So it is access without appropriate safeguards in place is incomplete delivery of a professional obligation. This is the battle we have been leading from WHO. And we have worked with big groups like Global Fund to say, look at your database, look at the number of proposals you have between round 4 and round 9 of your proposals that you are receiving for procurement from the countries. Do they have pharmacovigilance? Some of them refer to pharmacovigilance in their proposals, but in practice, they were not following them through. It was just one more clever way of getting money into the country, but diverting the use of that money, not for pharmacovigilance, but for something else. So now, of course, Global Fund has been listening to us, and we have the opportunity to introduce, through the Global Fund grants, minimum pharmacovigilance requirements in the grant implementation countries, and we've also set up a toolkit that countries can use while introducing pharmacovigilance. So, now to the bull by the horn, biotherapeutics. You know this better than I do, that they are generally large, complex molecules produced from living cells using biotechnology, can pose rare but serious risks of unwanted immune responses. We've been hearing about this morning, yesterday. Require extensive testing and risk management planning. Nothing new. You've already heard that, you know that. Coming to similar biotherapeutic products, SBPs, as WHO, I often say, I think we chose SBPs because WHO likes acronyms, and SBP is an acronym we can do. We like acronyms with 3 letters. So SBPs, copies or generic versions of biotherapeutic products. These are not exact copies of innovative products because of the manufacturing processes involved. Unlike other generics, we cannot rely on data for or from innovative products. Therefore, it needs sufficient proof of similarity to innovative product, both preclinical laboratory testing and clinical trials. You know all that. Risk management plans are also essential. I will not talk about this because I have Niels who's going to, and others who are going to cover it. Guidance from WHO, you already heard from Ivana, developed in 2009, published in 2010, based on the EMA guidelines of 2006. Pharmacovigilance of similar biotherapeutic products. In this guideline, it is mentioned very clearly that the manufacturer is required to submit safety specification and pharmacovigilance plan at the time of submission for approval. Safety specification should describe the important identified or potential safety risks for the reference biotherapeutic product and the substance class and any specific safety issue that is specific for the similar biotherapeutic product. This is clearly mentioned in the guideline. And the risk minimization activities that may be needed should also be presented. What is intended? What will be carried out? What are those activities, including whether there would be education material for patients, for the healthcare professionals, etc. The post-approval activities for the similar biotherapeutic products, due to the potential of the SBPs to provoke immune reactions, the manufacturers will have to undertake post-marketing surveillance to the same standards. So this is where the clear difference from the generic traditional chemical medicines, okay? So it has to be of the same standards as the reference biotherapeutic products marketing authorization holder. And the pharmacovigilance plan should include safety monitoring as required of the corresponding reference product, safety monitoring for additional risks that may have been identified with the similar biotherapeutic product. And the similar biotherapeutic product manufacturer is required to have pharmacovigilance systems in place, just like the reference products. At the time of approval, there should be a qualified pharmacovigilance personnel associated with the system, and there has to be a means to report to the national regulatory authorities where the reactions occur. So it's all there. Now, the next step would, of course, be to walk the talk. So it's all there. The next step would be, how are we going to see this implemented in practice? Traceability of adverse events. It is an issue for everything. It has been mentioned, and you will hear a bit more about the names session later. But adverse events may be product specific. So we have to be able to make a correlation of the adverse event to the product. Therefore, in any report, whether it is any medicine, not just a biotherapeutic, any medicine, from the WHO pharmacovigilance program, we've always been emphasizing that critical information that will help us assign the adverse event with the specific product, be it a regular medicine, be it a biological, it has to be there. And any adverse drug reaction report for any product should include the INN, the proprietary or the brand name, the manufacturer's name, the lot number, and the country of origin. We have said this for the last 40 years, but the similar biotherapeutic products will give us an opportunity to reinforce this. Now I thought I will just quickly share some data out of our database. And I will not be able to give you a lot of intelligence into the data that I'll share with you because I got this data the day I left. So I haven't really been able to query the information from the collaborating center in Uppsala, but they were kind enough to expedite the search. They were on holidays on a long weekend, so I wasn't able to contact them. But this is what it looks like. The number of reports per million inhabitants per year. And you will see that most of this information is coming from advanced settings. Cuba somehow made it there. That's good for them. But this is the number of individual case safety reports per million inhabitants in the country per year. And this is what we have seen in the database the last 5 years. This is for all medicines, okay? And then I asked this question. I just said, randomly I picked up Let's look at monoclonal antibodies, because I couldn't have them do everything. There are 8 million reports in the database. So you can imagine that they aren't jumping with joy when I give them a last-minute request. So reports on monoclonal antibodies, I said, for want of anything better. And this is the number of individual case safety reports that are there in the database, 350,000 plus. And out of those, how many are those that are referring to INN, trade names, and indications? More than 2/3. So that's a good sign. But of course, these are all innovator products, right? So the tendency is there. We have to make sure to carry it to the SBPs. If I would then break it down to distribution of reports of those monoclonal antibodies and take out the top 10, who is reporting the most? Anybody's guess? It is the United States of America, of course. And moving on, if I would say, what are the top 5 adverse drug reactions with these monoclonal antibodies in the WHO database? You can see for yourself, 18,000 plus, 15,000 plus drug ineffective. I think that's very interesting for me. 15,000 reports of drugs not being effective. What I'm trying to point out here is that good data in would allow us to make very good assessments. It is possible to pose all kinds of queries of the data that will tell us better what works, what doesn't, what kind of risks we need to watch out for, and how to position ourselves and prepare ourselves for it. So, and if I just thought it might be interesting for the participants to look at this data for countries in the region. And you can, for the monoclonal antibodies, the number of reports in the global database is what it looks like here. And Russian Federation has some 23 reports. So I will say that the similar biotherapeutic products, let me just restart. If I would say that today we have 8 million reports in our database, most of those reports are coming from stringent settings. Although we have 146 countries with pharmacovigilance systems in place, most 50% of our data comes from North America. So it really is as if the rest of the world is not contributing very much to the database. The problem has been that though we have systems in place, the capacity to operate isn't there. So my opinion is that, in my view, occasions such as the biosimilars give us an opportunity. So let us use biosimilars and other products as entry points to get into the country, work with the countries, to put a system in place that will perhaps address safety issues related to biosimilars to begin with, but you will create a system working for the biosimilars that will then be able to take on the safety monitoring of additional products. That's what I'm saying. SBPs provide an opportunity to engage in private-public partnership, PPP, for pharmacovigilance in low and middle income countries. We need to be working better with industry. We need to be working better with countries. And countries and industry need to be working better together, because it can be a win-win if you will all play our cards well together. Because the manufacturing authorization the marketing authorization holder of generic and innovator biotherapeutics are both obliged to invest in pharmacovigilance. It's not just the innovators anymore. Both are obliged to invest in pharmacovigilance. And as you know, generics are often used in low and middle income countries. And low and middle income countries are those places where there is no pharmacovigilance operating really. So therefore, by introducing and through pharmacovigilance of SBPs, it is possible to create pharmacovigilance systems in low and middle income countries or strengthen pharmacovigilance systems in these settings that will be used not only for SBPs, but also for other medicines. Governments and private industry need to work out a model where the data on products will benefit both the marketing authorization holder and the regulator, and the pharmacovigilance system that the two groups will develop together for SBPs will lead to capacity in the country. As they say, I saw this in one of my travels in Ghana. They said Rome was not built in a day. In fact, this shop was called that. I loved it. Rome was not built in a day. I think we all know that. And so, step at a time, we do need to build structures and best practices because, and I just have to make this quote from one of my gurus in pharmacovigilance who happens to be sitting here in the room, Professor Vladimir Lepakin, because he said, he was our Assistant Director General some years ago, and he was also a lead in pharmacovigilance, and he said, dying from a disease is sometimes unavoidable, but dying from an adverse drug event is unacceptable. I think it says everything. So with that, I thank you for your time.
Roman Ivanov, PPR&D, Biocard company. I just have a maybe technical question. Do you know which approximately share of adverse events in the WHO database comes from marketing authorization holders, I mean, on regulated markets, of course, and which part comes from medical community, from healthcare providers?
I couldn't answer that top of my head, but it should be possible. But in general, in our database, global database, we never receive reports directly from the marketing authorization holders. They come through the pharmacovigilance center in the country.
Okay.
So, yeah.
So it's a pity because it would help to answer the question who is to blame, I mean, to whom place the blame on the so underreporting of adverse events on, I mean, non-regulated markets, whether, I mean, originator companies do not report adverse events as they do on regulated markets, or it's healthcare providers who do not participate in pharmacovigilance programs.
It's my guess that we all share the blame.
Okay.
I don't think that we can just point to one group. I think we all share it.
Thank you. And another small question. It's an interesting proposal to build a pharmacovigilance infrastructure in the company using biosimilars as an example. But don't you think that the frequency of unexpected adverse events is, I mean, is much higher? I mean, the risk of unexpected adverse events is much higher in case of innovative biologicals that are with novel mechanisms of action that were, I mean, just completed their registration studies. I mean, and the risk of something unexpected is really higher in that example in comparison to biosimilars with very well-known mechanisms of action if they, of course, followed the, I mean, suitable registration pathway through comparability with the innovator.
I think you've answered if they have followed suitable processes, yes. And the data that has been referred to so many times in the last day, you know, that if they have met with the requirements, yes.
So if the biosimilar is studied in a proper way in compliance with international guidelines, you think that the risk of, I mean, adverse event probably is actually lower than in case of novel drugs with, I mean, new mechanisms of action, right?
I wouldn't, I wouldn't, that is speculative. That is speculative.
Okay,
and I don't think we have the luxury of speculating in health.
Okay, thank you.
And how it correlates with change in market of pharmaceutical drugs, especially the emergence of biosimilars, biological products. First of all, our system in Russia has quite a long-lasting history. It was initially settled in middle 90s, following the models provided by WHO. Also, I would like to dwell on some issues, aspects of the way this system looks today. It was legally framed in 2002, and principal elements typical for national system of pharmacovigilance were created based on collection of spontaneous reports. This is hypothesis on safety, drug safety signals. There's our legal regulations shown on this slide, stipulates or states specific key duties for medical, for all those involving medical products circulation. First of all, stakeholders have to inform us of all events that take place, predominantly unexpected events, which are serious, unforeseeable, unexpected. This is first of all based on the safety profile of the drug, and then periodic reporting, two-stage reporting. First stage is the first registration of the drug, and before, based on standards, periodic reports on safety profile, and finally, punitive sanctions when players fail to provide all the safety data on the drug circulating in the market. Our system is oriented on electronic collection of data, spontaneous reports from physicians and those who develop their drug, also sponsors of clinical trials, and then we analyze these data in our organization, expert organization. And finally, we build up recommendations that are considered afterwards by our healthcare ministry. And based on this consideration, our ministry makes regulatory decision. So, we are analog of SHMP, while regulatory function of Euro Commission is addressed to the Healthcare Ministry. Since a large number of Russian manufacturers or enterprises have not transferred to GMP yet, we still have problems with storage of medicinal products. Then, part of our work is devoted to quality control and also inspection of the issues associated with quality of the drugs on which we get information. So, this is specific feature of Russian pharmacovigilance system of safety monitoring of medicinal drugs. I'll omit some of the formal quotations from our legislative acts. Only one thing is important: administrative sanctions. Sanctions in respect to manufacturers. Such sanctions are not enough because they are not really restricting so that to provide 100% performance on reporting. But hiding of important information on safety that would require changes in the leaflet, in instruction, would result, for example, for withdrawal of registration. Our decisions are harmonized with the old version of European legislation with the time frame existing before introduction of guidelines on GVP, guidelines of GVP. There are some differences in dates in our practice. 2 months. 1 month after the period of data collection is over, terminated as distinct from 2 months accepted in Europe. We are tolerant, nevertheless. We actually accept reports in 3 formats, starting from older guidelines, ICH E2C, a classical format, up to amended, updated periodical report on benefit-risk ratio after the second review of these guidelines. And finally, we are going to present the last update of this version of the guidelines specifically for Russian manufacturers, so that to give them maximally detailed explanations on how a quality report should be prepared, how to work during the data collection period, so that the collection data is really sufficient. Our work during last 4 years allowed us to improve the reporting on adverse reactions. Also, we cannot view it as ideal. It's too low, twofold, let us say, less performative than the similar work in the United Kingdom. From 2009 to 2010, we observed the increase in the reporting. The providers, key providers of such info are the providers of medical services because we offered very tool, instrument to provide these reports via internet. And thus, we get an increase in reporting, which is approaching to the phase of plateau. And such a phase requires quite different mechanisms of reporting. It's time really to engage factors that identify the insufficient number of reports typical for or underreporting typical for low-income countries, described in the classical article of Inman. Finally, regulation of the activities of the performance of manufacturers on collection and assessment of spontaneous reports, and also introduction of good practice, although sanctions would suffice, they will provide adherence to these requirements. It's also important to underline that pressure, administrative pressure on physicians, such as an attempt in Italy to introduce criminal liability for hidden info, it didn't bring about better reporting. And even if we get better reporting after such pressure, we'll start getting typical reports, senseless reports, just an additional workload, but better efficacy in our performance following these events. What else is important? This method is the most effective, and we formed a group team of well-trained specialists oriented at identification of such reactions that are familiar with the particular range of medicinal drugs, and those people who are really able to report to write such reports. Many of them have become key elements in identifying new safety issues in biosimilars, such as it happened with erythropoietins or monoclonal antibodies and opportunistic infections associated with use of MAVS. And however ideal is the methodology of spontaneous reports, there are particular limitations, certain limitations that are quite obvious to all of us. They deal with the generation of hypotheses but not confirmation of safety signal. Second issue is the sort of correlation between mass media and the number of reports. This is also one of the limitations in our work, because maximum number of reports are associated with new drugs emerging in European, American market, where we always observe better reporting of adverse reactions of events. Thus, statistically, the probability to get safety signals in those settings is higher. Thus, we associate our underreporting with monitoring of scientific press and also foreign regulatory decisions. And what's important, we cannot identify the incidence of adverse reactions while assessing, analyzing spontaneous reports, because we never know the exposition to, exposure to the drugs, and we never know the exact rate of the reporting, and we never can compare the reaction to original drug and the generic, and we cannot monitor the kinetics of the incidence of adverse reactions and also decrease in efficacy of drug. And all these differences are of key importance for proper adequate control of safety of biological drugs in general and biosimilars in particular. And I'll cover that issue in the second part of my presentation. The world changes, and thus patents are expiring. Patents for the first group of biological drugs. Moreover, we should not anticipate improvement of this setting, because from 25 to 30% of new drugs in development that are developed by transnational companies, they are biological drugs. The population in developed countries is getting older. Medicine is becoming more and more expensive, and thus access to non-patent versions of earlier biological drugs is really necessary. So, as the rest of the world, we are also arriving to compromise between science, ethics, and financial issues, economics. That has been decided about 10 years ago as related to generic, but today it's a different issue and also more significant, because biosimilars that are similar in between each other as the dogs of the same breed. They are as different cubs in the same family, they behave differently. So, we bring them to the market, and puberty of such a cub arrives much later after it was given a marketing authorization. And principal difference between biosimilar and generic is the difference between the gadget of 20th and 21st century. Just imagine Victorinox company that was selling knives that were so popular, and now these knives are replaced by iPhones in the pockets of young men. The company was not obliged to monitor efficacy and safety of that particular gadget, whilst currently, when a company sells modern accessories and gadgets of pharmaceutical companies or any other working at the market of innovative technologies, they also sell services. And this cycle of relation of a seller and buyer is not limited by this stage. So, the complexity of biosimilars elongates the process of studying their safety through the whole life cycle of biosimilars. It was studied with the emergence of the most complex drugs on the pharmaceutical markets in 80s, 90s, when interference beta emerged. In Russia, for example, first recombinant drugs such as interferons emerged in 90s. And all the same story with insulins. And today, with introduction of good pharmacovigilance practice, this process involves the whole life cycle of biosimilars in Europe and has become one of the elements of good practice actually. So, we have to accept a compromise or the second version of the generic compromise with biosimilars. Of course, we cannot issue a biosimilar. We keep tossing over this issue for a day and a half already. In the United States, the latest guidelines recommendation, the access to the markets is based on limited preclinical and clinical studies, pharmacodynamics, pharmacokinetics, and toxicity studies of biosimilars. And besides, in some limited clinical studies, comparative studies with on one of the indications. Because we are not always sure whether we can translate these data from one indication to another with biosimilars when comparing with reference drugs or originator drugs. Thus, we have to deal with surrogate endpoints such as the control of the focal lesions on MRI to evaluate the efficacy of systemic sclerosis, motor activity of the patient, for example. They are surrogate endpoints. In countries that accept this concept, they are nevertheless key issues for each biosimilar. First of all, this is absence of data about its long-term immunogenicity. I am speaking of a typical for all biosimilars problems or challenges. Again, we have to take into account all safety issues of the originator drugs. They always exist and require same control methods and risk minimization activities. This all refers to immunogenicity. Each time when we change the manufacturing process, the issue emerges anew. And even if the drug is not really immunogenic, antibodies does not perform as neutralizing antibodies. Very small percentage of them are present. Nevertheless, we should identify the populations of patients in whom that therapy would be ineffective, and each time we have to provide very specific methods, cheap methods of antibody formation in those populations. And then, the necessity of additional interventional investigation studies so that to translate data about safety and efficacy of the originator to a biosimilar, we cannot allow automatic use of a biosimilar on all indications for a significant proportion of biosimilars at least. And finally, it's the issue of interchangeability, replaceability, etc., and quality control issue. In the whole world, the union of pharmacovigilance and regulation got different programs on risk management, different names. RAMS risk regulation, mitigation strategy in the United States, and plan of risk mitigation and risk management in European Union, which is prepared at the pre-application stage. And it covers the adaptation strategy of investigation studies, specific for each market, risk management tools, and the key role is with observational studies always. In some cases, we can perform prospective studies, but in most cases, biosimilar cover large populations of patients and they are used as the final drug when we cannot collect, accumulate the sufficient number of patients, so we should start the registers to get sufficient data on nature of them. So, I want to draw your attention to two slides of Boris Romanov. You see them now? These slides demonstrate the most typical methods of pharmacovigilance for identification of each time of reaction, specifically effects with long-term use and also long-term side effects as cancerogenicity cannot be covered by spontaneous reports. And starting from the specific of those methods that are covered by ICH guidelines within the pharmacovigilance, it is clear we have to introduce pharmacovigilance programs in post-registrational phase, but we should plan such programs before the application is submitted. So, we see the necessity to improve the pharmacovigilance per se and regulations as well. Following this, we provided the drafts, amendment drafts to improve some articles in the federal law on drug circulation. First of all, this is harmonization of basic definitions in pharmacovigilance, such as adverse reactions, serious adverse reactions. We should harmonize it with ICH so that we could perform similar spontaneous reporting in different countries. Second issue. We also propose that manufacturers could resume their research programs following the requirements of regulators because it's of key importance that all functions are in place to mitigate the risk and minimize the risks. And these are the tools of safe presence of the drugs on the markets. But what's again important? Despite the fact that our legislation in pharmacovigilance and also measures to manage the risks are not in place, it doesn't mean at all that manufacturers of biosimilars does not monitor safety and does not try to mitigate the risks of the use of the drugs. Important is the fact that these measures are mandatory for all manufacturers and are continuously used. Modifications in the market, changes in the market, emergence of monoclonal antibodies, there's a pause in their translation, there are just two words that allow to conclude. 3 words to conclude. We are never jobless, and to adapt to new drugs, specifically to monoclonal antibodies that are quintessence of all biosimilars, we have to work further, and from one side we have to provide very strict, strengthened rules in pharmacovigilance. Inside the companies, we should provide tools for reporting with electronic support for the acceptance of spontaneous reports, collection of spontaneous reports. We work with the WHO to introduce here the auxiliary system of registering of finance reports. And again, we strengthen sanctions within the pharmacovigilance, which should develop regional segments of pharmacovigilance to form these very functional teams. And we have to provide safety and to study particular issues and challenges, And finally, we have to improve cooperation with international regulatory bodies and international organizations so that properly data that are collected globally on biological drugs and particularly on biosimilars are known also to enhance our national export potential.
Спасибо, я готов ответить на ваши вопросы.
Thank you, Sergey. So that was interesting to listen to the growth of the program, the reports, some of the underlying causes on the number of reports going up, and how health products are not the same as gadgets that are sold in markets, and how we can learn from dogs. So that was good. And I would invite a couple of questions on Sergey's presentations. Yes, we see. I kindly introduce yourself and ask you questions.
Пожалуйста, Боинг, компания Sanofi. Сергей Владимирович, будь добр,
Sanofi company. Sergey Vladimirovich, please tell us about the role of the patient organization that are now involved into the monitoring program following the amendments, drafts to amendment to the law on drug circulation. What are their liabilities? What are their rights of those organizations? So what was the idea behind their involvement into the process?
Federal law, that is why I cannot comment in detail the content of the document after it was submitted to the Ministry of Public Health. But what I would like to mention, first of all, we should expand. First of all, the should take more and more reports from patients, because the European regulation is when you evaluate the benefit-risk ratio, you always take into consideration the opinion of the patient. When you filter this report through physicians, then we lose some info. Some facts are hidden, some info, some data that patient considers very important, such as irritation interfering with therapy, that are important for the patient, they are ignored by the doctor. That is why we're trying to work with the patients and we try to react to the signals from patient. But as for today, I cannot tell you in detail how we are going to regulate the participation of patient and patient organization in the monitoring process. I can guess that the role will be similar to the role that is given to them in the regulation of safety of drugs in programs that are in force. Nazarova, asking a question. So we have new biosimilars emerging into the market. Are there any specific actions, if there are any, that you deem to undertake? Companies, Roche, Merck, Glaxo, any company. And for example, they have 500 reports on the original drugs, and then a new company comes with a biosimilar and with no reports on a biosimilar. Not a single report on adverse events during 1 year. So you know all those things from new companies coming into the market. So do you envisage any statistical investigation and sanctions probably, because it's really impossible if you have 500 reports for an original drug and no reports for biosimilar. Heading of the N4 with synonymic replacement is not a case at all. Sometimes, on the contrary, we have some spikes of reports with biosimilars. Another issue is the quality of those reports. Of course, we should implement sanctions when there is underreporting and hidden data, but we are going to implement them only when there are standards covering the performance of the companies. As for today, this mechanism can be implemented only in case when we know about the identified reaction in a medical setting, when the doctor reported, and in parallel the physician, the doctor, also reported to the company, and company tried to hide this report. But nevertheless, we need a specific standard for reporting, specific requirements to acceptance procedure and monitoring procedure, and thus availability of legislative standard envisages also the chance to inspect, to audit. Otherwise, we cannot deal with underreporting of the industry, of the manufacturer. There are no other tools to control. Kudina, Novartis Pharma. My question deals with sanctions and actually situation with underreporting. You mentioned in these underreporting in your legislation up to withdrawal of registration license or marketing authorization license, and if company underreports with the specific requirements from Roszdravnadzor and company does not adhere to the requirements. So what should be, what is the solution? Now we have to apply to the court, and then arbitrary court would actually impose sanctions on the company. Are you familiar with such cases? Well, it's just the beginning of the story, and currently we can impose penalties directly on such companies.
Okay, good morning everyone. My name is Niels Vermeer and I'm working in pharmacovigilance at Utrecht University and the Medicines Evaluation Board in the Netherlands. And my talk will be about risk management plans within the life cycle of management of biologicals. My presentation will actually comprise two parts. I will first give you an introduction about the key features of the risk management plans in Europe. And the second part will be about a research which I'm currently conducting into the functioning of the RMP in Europe currently. With the following researchers we are listed here on the slide. This is a disclosure slide from the Utrecht University, and I need to point out that the views expressed are my personal views only. Well, the risk management plans were introduced in Europe in 2005 to strengthen the coordination of the ongoing safety assessment and risk minimization after a product has been put on the market. The reason for introducing this risk management plan was that the safety data from clinical trials, the limitations thereof was increasingly appreciated. These limitations include limited follow-up of participants, strict inclusion and exclusion criteria, and, well, most importantly, a limited number of study participants in the trials. This was actually recently published in PLOS Medicine, showing that on average 1,700 patients are exposed to a product before it is put on the market. This was over the last decade in Europe. And other reasons for introducing the RMP or the risk management plan were high-profile safety withdrawals, for example, rofecoxib, Vioxx, due to cardiovascular disease. And also there has been an increasing pressure over the years for earlier access to new medicines, both by pharmaceutical industry as well as by patients. Well, the idea of the risk management plan was actually first proposed by the Japanese Ministry of Health and Welfare in 2001, after which the ICH guideline provided further backbone to support further guidance. And well, the rationale was clearly stated in the concept paper which they drafted in 2002, stating that, well, carefully planned pharmacovigilance activities can reduce the risk of drug toxicity and, very importantly, can help avoid withdrawal of effective drugs from the market. Well, they also stated that, well, pharmacovigilance activities should be planned before a license may be granted. And, well, this is the case in Europe since November 2005. Biological may only be put on the market after RMP has been put in place. The requirements, therefore, are described in the guideline for good pharmacovigilance practice. And, well, the RMP actually comprises three main sections: the safety specification, the pharmacovigilance plan, and the risk minimization plan. I will go into detail on this section in the coming 3 slides. At first, the safety specification is a sort of synopsis of the safety profile on basis of the non-clinical and clinical data gathered so far. And furthermore, it also summarizes, for example, potential for off-label use or potential for medication errors in clinical practice. Within this safety specification, 3 types of safety concerns are distinguished, which are identified risks, for which there's adequate evidence of an association with the product. There are potential risks for which there is a basis for suspicion of an association, for example, based on a class effect or based on non-clinical data but was not confirmed in clinical trials. And lastly, there is important missing information, which is safety data which is not available at the time of the registration. For example, safety in renally impaired due to the exclusion criteria. Previous research from Giesen et al. In 2009, you can see it on the table below, showed that safety concerns for biologicals are more frequently classified as missing information and less frequently as identified risks as compared to small molecules. This indicates that, there is a greater uncertainty on the safety profile of biologicals at time of approval as compared to small molecules. The second part of the RMP is the pharmacovigilance plan, and it's actually a plan to further identify or characterize the safety concerns which are described in the safety specification. For each safety concern, the need for additional pharmacovigilance activities is determined. This might be additional clinical studies, registry studies, non-clinical studies, and so on. And it might also be the case that routine pharmacovigilance is considered sufficient, so collecting of ADR reports and providing regular updates to the authorities. From the same study in 2009 by Giesen et al., it was also shown that for biologicals it was more frequently required to have additional studies put in place after marketing authorization as compared to small molecules. The final part of the risk minimization plan of the RMP is the risk minimization plan. And again, for each safety concern, the need for an additional risk minimization activity is determined. This can be an educational program or a controlled distribution. And it might also be the case that routine risk minimization is considered sufficient, and that comprises just labeling in the normal product information. On the bottom you can find an example of an additional risk minimization measure for eculizumab. This product was associated with an increased risk for severe meningococcal infection, and therefore patients are required to carry this card at all times to warn any attending physician that the patient uses this drug, and once the patient develops symptoms of this infection, immediate treatment should be initiated. When the RMP has been finalized, the summary is published on the website of the European Medicines Agency, and it looks like as shown here. So for every safety concern, the pharmacovigilance and risk minimization activities are summarized. Then after marketing authorization, the role of the regulatory authorities does not cease. There's nowadays a continuous interaction between regulators and pharmaceutical industry, and so there is a continuous benefit risk assessment. This means that the risk management plan should also evolve as the safety profile becomes further defined, as more knowledge becomes available. However, we do not have any insight of how this RMP evolves, and how the activities proposed in the RMP contribute to getting the required knowledge earlier than only waiting on the spontaneous ADR reports coming in. So thereby I come to my second part of the presentation. So the study aim of us was to describe the evolution of the RMP after marketing authorization. And this can provide insight into the knowledge gain of medicinal products over time and how the RMP functions currently in Europe within the life cycle. Our secondary aim was to study factors associated with change, in particular with respect to the additional studies proposed, because this can provide insight on how these studies help in gaining the required knowledge earlier. We are therefore conducting a cohort study. We include 56 new medicinal products, and we follow them up until the end of 2012. And the outcome of interest is a change in safety concern. So any baseline safety concern can change after marketing authorization, or can be omitted from the RMP when sufficiently studied and not considered to impact on the benefit-risk profile. And also, safety concerns may be newly added. We received all RMPs and updates through the European Medicines Agency, and we extracted data in a longitudinal manner to allow our data analysis. Now the results of these 56 products, 23 products concerned biologicals, and we were ultimately able to include 17 of these into our studies. We had to exclude some products because they either concerned vaccines or they concerned cell therapy, and we had to exclude products with no RMP at time of approval or with no follow-up, at least not to our knowledge. So these 17 biologicals contained 253 safety concerns. So on average, 15 concerns per product. And we follow these 253 concerns up for change after marketing authorization. You can see that the median follow-up was approximately 5 years, comprising a median of 8 RMP updates. This shows how the safety profile evolves after marketing authorization. On the left, you can see the baseline safety concerns. On the right side, the change. And you can see that, 19 potential risks changed to identified risks, and 20 safety concerns were omitted during post-marketing phase, meaning that they were probably sufficiently studied and, could therefore did not require any longer follow-up as part of the RMP. The reason for omitting this safety concern was in 9 cases resulting from completion of the pharmacovigilance activities proposed in the RMP. So in these 9 cases, the studies proposed really got the required knowledge which was lacking at time of approval. 3 resulted from completion of studies outside the RMP. For 4, there was no new data, so probably was considered that the potential risk was not considered identified after all, so as no new data came in. And for 4 cases, for 4 safety concerns, we did not have any information on the reason. This slide shows you the timing of safety concerns being omitted. On the horizontal axis, you can see the time since marketing authorization in months. And you can see that, the first safety concerns were omitted approximately 2 years after approval, and most of them are omitted after 5 years. So, this gives you an idea of how fast the information is coming in. Finally, 59 new concerns were added post-marketing, and the data sources were cited as clinical studies, spontaneous reports related to new indications with new comorbidities, and so on. To conclude, risk management plans can play a very important role in the life cycle management of biologicals, and they can support ongoing benefit risk assessment post-marketing. Well, the European guidance has provided the backbone here for, well, this is an integral part of the regulatory approval of new biologicals. And also, well, regular updates are provided post-marketing. As we saw in the study results that well, the observed development of RMP supports their role in the life cycle management. However, the vast majority of the baseline concerns remained unchanged 5 years post-approval. This might be caused by the fact that there was only a not very long follow-up, only 5 years, so the safety concerns might not be sufficiently characterized at that point in time. And also very importantly is that, well, it's very difficult for regulators and pharmaceutical industry to decide that a safety concern needs no longer follow-up as part of the RMP. It's always safer to say, well, let's keep it in the RMP for another 5 years and omit it after that, after we do not receive any new information. Well, to conclude, well, 5 years post-approval, the emphasis of the RMP, the involvement seems to be on newly emerged concerns rather than on changes in baseline concerns. With that, I would like to conclude and thank you all for your attention.
Thank you, Neil. I invite some questions, comments. I see a hand go up here, and then another.
Niyazov, Scientific Center for Evaluation for Medicinal Products, Ministry of Health. I'll give my question in English first and in Russian, so there is no need to translate it.
Thank you.
According to new pharmacovigilance legislation in EU, in particular Directive 2010-84, which amended Directive 2001-83, competent authorities entitled to oblige to conduct post-authorization efficacy studies. This is a very important issue, especially in the light of approving some indications for biosimilars without any clinical data. So my question, what is the experience of the EU competent authorities with assessment such data?
Well, thank you for your question. As you rightfully pointed out, this is a very recent requirement. And to be honest, I personally do not have any experience with plans for efficacy studies being submitted. I was recently involved in the assessment of an RMP for a new product and did not make part of the RMP, so I cannot answer your question, unfortunately. Maybe.
So post-authorization efficacy studies will not be required in all cases for European centralized products. They will be required in cases when there is need to generate additional interventional data in missing part of indications or part of subpopulations where it's regarded necessary for prescribers. There is only limited experience at this moment with biosimilars. It's thought to be not necessarily the area where the studies will be often conducted. It will be most likely that originators companies will be likely to conduct quite a lot of those additional efficacy studies. For those extrapolated indications, the area might be covered with some observational safety studies, and there is experience with one filgrastim product and with erythropoietin product, which is actually been based on some post-marketing studies.
Thank you. I see a question to my right, yes?
My name is Jaap Veem, I'm from AbbVie in the US, and I could ask my question in English and Dutch, but that would probably not be very effective. My question is concerning your data in your study. I would argue from a public health point of view is that the inclusion of previously unidentified risk is actually more important than the omission of risks that are potential but do not emerge during pharmacovigilance. So my question is, does your data show a difference in timing as to the identification or the inclusion of new risks? Does that take longer or shorter, or do you focus on that more than, for example, omitting a potential risk from a pharmacovigilance plan? Does your data show this, or do you have an opinion on that?
So you mean as compared to safety concerns being omitted? Is that your
Yes. I mean, I think a risk that was previously not identified, but actually emerges in the post-marketing period, is more important than a risk that may be potential, but ultimately does not prove to be a risk, because conceptually that's more important for public health, I would say.
Yeah, sure. Yeah. I agree that it's really important to also have these newly emerging risks post-marketing, to include them in the RMP. But we were actually also very interested in how the RMP contributes to gaining further knowledge on this missing information. So that's why we primarily focusing now on the missing information being omitted or becoming identified post-marketing, as this really shows us what the influences of the RMP on gaining further knowledge. But I do agree that we should also look at the newly emerging risks and do a time trend analysis on this. So, thank you.
Okay, I'm quite sorry. But again, like yesterday, I'm standing now between you and lunch, and now we even have crossed the border. But I hope you can keep up for a couple of minutes. I really will do my best to make it quick and focus on what I think quite some important slides only, because you have heard a lot before already. I also have to correct the chairman. I'm not speaking on behalf of Roche, this time on behalf of IFPMA, the Biotherapeutic working group we have there, and I'm a member of this as well. So in fact, this reflects a more consolidated opinion of this group here. So let's keep the content. I think we discussed this already quite in detail. If you look on a global level and you compare pharmacovigilance practices in different countries, you see incredible differences. The point is very clear, and the speakers have pointed this out before. We really need to strengthen the system, and that means, in many cases, to establish pharmacovigilance laws, eventually to provide proper systems for naming, to increase the reporting and the collections actually of these events. And as in Europe, this is now also going on, really focus a bit more also in the context of the benefit actually of these drugs. Here you see what is in fact going on in Europe since 2010. So there is a huge effort, in fact, strengthening the monitoring, also clarifying and simplifying the tasks, improving decision-making procedures, because as you know, I think one thing is collecting data, but then actions have to follow, of course. Strengthening also communication and transparency with respect to that. And I think in the presentation of Niels before, you could see already about the reporting, how this is presented at the web pages there and so on. Let me spend a bit around this slide, because in fact, I have to say I like it very much. Because from my perspective, this nicely shows you what is the core around pharmacovigilance, why this is so important. And here Thomas, that actually generated the slide, he made a tremendous job to, put complex things in a very nice visual way. So think about it. If you establish safety and efficacy, and it doesn't matter whether we talk about a biosimilar or whether we talk a new biologics, you do this in a highly controlled environment. And then you go out, you broaden eventually a bit your population, controls are less. And this is why we have to follow up. What is the effect, What does it mean for the risk-benefit of the drug, if we are, leaving the comfort zone of a clinical trial environment with our drug. Not to speak about if we are talking about off-label use, for example, another thing that is going to happen. But we have to collect all the data in order really to establish a proper knowledge management for our drug. Because at the very end, pharmacovigilance, it's also about knowledgement, increasing knowledge, increasing the database we have for our drug. Now, very quickly about a couple of things focusing on biologics, why pharmacovigilance is important for biologics. And actually it was very interesting also to see in the presentation we had before that in fact, the concerns may raise for biologics once they are on the market. I think there are also other studies around this that in fact goes in the same direction. So for sure, this is related to the complexity of the drugs. I think very clear. We can characterize all of this to a certain extent, but there are limitations. And at the very end, also in the context of biosimilars, we are talking about risk mitigation. On the one hand, you want to make it possible that these drugs get developed, because it's important that we have biosimilars. On the other end, you cannot, go forward with requirements that make the development practically impossible. So you find compromises there as well, allowing the development, within a reasonable investment. I think this is what we are talking about, but still we are having this complexity. Immunogenicity, we were talking about this before. All these biologics have or elicit an immune response. We will see anti-drug antibodies. And here in the lower table, you just see a couple of numbers of different products. But in reality, the situation is even more complex. These numbers you see here, you can split them off. And then you see in each and every patient population, you have these biologic as a treatment, a different immune response, dependent on co-medication and other patient factors. So it's really quite a complex thing we are talking about here. We are doing a lot to minimize, in principle, immunogenicity with the antibodies. We've heard this already this morning, going from pure murine sequences now to fully human antibodies. But still, even with the fully human antibodies, you see immunogenicity rates of 30, 40, 50%, dependent in which population you are looking at. About the consequences, we also were talking already to a certain extent. In the best case, you may have no consequence. You have these anti-drug antibodies floating around, but they don't interfere in any clinical outcome. But there might be consequences. There might be consequences for safety. This morning we were talking about PRCA, in that case neutralizing of the endogenous counterpart, but also consequences for efficacy. And let me talk about this as well, also again emphasizing the importance here. If we are talking about EPO treatments, you may measure easily, that the hemoglobin level is not raising. But now think about oncology. We all know that in many cases we are talking about a progressive disease, although patients receive a treatment. If you there get an antibody response, an immune response, as a physician, you will not be able to distinguish between the natural progression of the disease or eventually the progression caused by an immune response. And this is why immunogenicity is really a very important factor. As pointed out this morning, it's a multifactorial problem, and I really want to draw very rapid conclusion. As I was commenting this morning, you find some literature where people are saying, well, immunogenicity might be related to some leaching products coming out of the stopper. You also may find some publication where folks are saying, well, this might be related to some aggregates we found in a drug product. But I've never ever seen a clear-cut study showing that this hypothesis indeed is true. And by study, I mean a clinical study. And this is really the problem with all these findings, because as pointed out this morning, it would not be ethical, in fact, to do a study like that. Also, what drives the importance of pharmacovigilance for biologics, exaggerated pharmacology. Well, what is behind this, We may neutralize a certain factor, for example, with a monoclonal antibody, but then this may have additional consequences. Eventually, we may not think immediately about it. So examples for this are tuberculosis, when we are treating patients with anti-TNF treatment, so anti-TNF antibody, okay? And so this factor does not only have, as we all know, a negative impact in patient, but also some positive impact, and you get rid of this as well, and then you may have an increase in tuberculosis in these kind of treatments, eventually not immediately visible. The PRCA case also, you know, you may question whether this should be there, but it shows the principle again. The antibodies were formed against the recombinant apoetin. But at the very end, what led to PRCA was the fact that these antibodies also were neutralizing the apoetin synthesized by the patient's body as well. So also here again, secondary effects, not immediately related to the therapy. Another very important point why we need specifically for biologics proper pharmacovigilance is really the very different classes of products, let me call it like this, we have on the market. On the one hand, we are having the innovator products. Then, in the best case, we will have similar biotherapeutic products, but on top of this, we have lots of other products that eventually were approved by different regulatory pathways. Especially for these products, we have quite limited information on safety and efficacy. But they are out there on the market. And not having a strong pharmacovigilance in place, really it will be difficult to find indeed arguments why they should not stay on the market. But on the other end, eventually patients are suffering because of their presence. And we know, we discussed this yesterday, minor changes will have impact to these products. Also, similar biotherapeutic products may have a different, slightly different safety profile from the originator. We do some limited studies in the beginning, but then during the market, eventually this may turn out as something critical. You don't know. Non-comparable biotherapeutic, clearly we don't know a lot about it, especially if they have been approved based on a generic approval pathway and just on the basis of an analytical comparison. Here you really cannot say a lot. This is also important and very important actually to consider when it comes to substitution. Think about these products. They easily may substitute a true biosimilar. They easily may substitute an innovator product, and you have no clue actually about safety and efficacy of these products. This is why we strongly believe that track and trace is extremely important for biologics. All these products I mentioned before, they also may be approved for different indications. So you may have an originator product that has 6, 7 indications. But you may have a biosimilar product that then has 3 or 4 indications. And eventually, on the same market, you may have then a non-comparable biologics that may have all indications like the innovator. And they are all administered to patients. I can tell you stories, colleagues of mine, he went to a Peruvian hospital, and there in the shelf in the pharmacy, they had originator product and non-comparable product. And do you know what happened? The patient got a mix of both, because this was just what was left on the shelf. How do you deal with this kind of situation? Track and trace, extremely important for specifically also for biologics. And I think in a very nice direction really goes the new EU requirement, where they clearly make the point that you have to follow the product starting from the prescription over dispensing through to the application, to really ensure that we know what is going on with the patient. This is also why I really have a certain issue with automatic substitution, which really also goes in the direction of a bit of an out-of-control system. Very often, automatic substitution is accompanied by prescription by INN, which we see, in fact, in many countries, and physicians are even forced to prescribe by INN, okay? But for pharmacovigilance, what does it mean? If you only report by INN, at the very end, if you have all these classes of products floating around, which all have the same INN, what kind of pharmacovigilance can you do? It's not possible. You are not able to track the products, and you are not able to follow adverse events. Also, an important point to consider is really the switching of therapies. You may switch from an originator product to a biosimilar product, you know, eventually during a therapy. But here it's important, do you then switch back? You know, if you do all the reporting, for example, by INN only, at the very end, what can you say? Which product was responsible eventually for certain adverse events a patient may have? And very frankly, for example, again, given all this variety of different products we in reality have on the market, you may end up with a class-specific recall or something like this, where although you may have nothing to do with the adverse events eventually observed in certain patients, but if you really cannot track, you know, you may make something more out of it as it may be in reality. And you, as a manufacturer, may be forced to recall your product, although you are not related or cannot be related with the event at the end. So clearly, INN plays a very important role for the pharmacovigilance, for the prescribing, and so on. Unfortunately, there is really limited control about the INN, because in fact it's up to the manufacturer to request an INN. It's not up to the INN committee to enforce an INN to a certain manufacturer. But I think for sure in the afternoon, I'm just looking at you, we will discuss more about it. But I think there are ways out of this situation, And this is also currently discussed, and I think it's really going in the right direction, because eventually having a clear INN differentiation between all products, be it originator, similar, and even non-comparable biologics will really bring transparency into the system. So this is, in fact, my last slide. I think pharmacovigilance is a very important part of any drug regulatory system, is an important part of any drug development, and it's really extremely important also to track, trace, and then, of course, analyze the data you generate in this system and draw the right conclusion. And with this, I'd like to thank you for your attention.
Thank you very much. Thank you. Roman Ivanov. Thomas, thank you very much for this wonderful and very emotional presentation. I'd like just to ask you, do you agree that the most important factor for ensuring a pharmacovigilance program is to know what trade name of the biological was prescribed to a patient? Is it so? The trade name and the batch number.
I can answer to. No.
Sure.
The most important thing is to have a unique identifier.
It can
this would be my answer. This is the most important thing. The question is, how do we get a unique identifier in all the different prescribing, dispensing situations we have around in the world?
Okay.
And eventually, the brand name is not the right instrument.
Well, but if we have registered a brand name and a batch number of a product that is prescribed to the specific patient, then for sure we'll be able to trace adverse events to a specific brand name, to a specific product. What my point is that, it doesn't really have to do anything with interchangeability of biologicals, because it doesn't matter, whether there is a differentiation of that step of prescribing. If we know that certain product was administered to a patient, then we can enforce the pharmacovigilance program.
I think having really a specific identifier available will facilitate things at many ends, because as pointed out before, you may have different indications for different products around. This also may then facilitate things in the hospital itself, in the pharmacy. I think it's very clear to everybody. Pharmacists, they work with INNs, this is their daily business. They are using this. You have the systems around prescribing by INN. Now, if you really come up, eventually with a unique identifier at this end, really for each and every biologics, this may make things very clear and very transparent.
For sure, I agree with you, but again, in terms of pharmacovigilance, the most important thing that we should know is what drug was administered to patient. And it doesn't have anything to do with interchangeability on the level of prescription. That's my idea.
Thomas, in support of this comment, I must share that our position in pharmacovigilance has always been that we will need the trade name together with the INN. So our reports in the global database, yes, we do solicit both. So I do have, I understand where you're coming from, but the key, as Thomas mentions, is the unique identifier. But we have, in the interest of assessment and analysis and having as much information as possible, we solicit both INN and trade name.
Good afternoon, Alexander Jan, Biotherapeutic.
my question is to Professor Miller. Yesterday there was shown a slide on which the humoral humanized antibodies should be more immunogenic as fully human ones, right? So my question is the following, two questions. Is it confirmed by pharmacovigilance that ADRs are bigger in chimera, humanized, if compared against fully human ones? And the second question, why, if immunogenicity is so important, the more immunogenic chimera and humanized could not push out of market the fully human antibodies? An example of CD20 fully human failed to push out of market detoximab. Thank you.
Questions. In fact, you know, around immunogenicity, it's not an easy thing and it's a multifactorial problem. The sequence is one contributor to immunogenicity. So whether you have a mouse sequence or whether you have a fully human sequence, this is one part of the whole picture. You cannot relate everything, you know, to these kind of sequences. There are many, many more factors are playing a role in this. And in fact, the interesting thing is, there are also recent publications that eventually certain sequences, and this has nothing to do with mouse or human, in the IgG1 itself may trigger certain immune responses or not, you know. So it's really, it's quite a complex issue. Now, why fully humans have not pushed away the chimeric antibodies. In fact, these chimeric antibodies, they are safe and efficacious. They have a history, you know, and in order to push them out of the market, you for sure would need something that is better. Isn't it?
Thomas, you suggested different INN. If you suggested even for highly comparable biosimilars that have thoroughly compared themselves against the reference product, then I would come back with a proposal referring to our discussions yesterday about manufacturing changes that the originator frequently do. Then I have to propose, if a manufacturer does a significant change to their product with proven changes in many quality attributes, as we saw on slides yesterday, then the originator would also have to have a new INN for their product if they do such changes. This is my counter proposal.
Okay, I was expecting this, of course. But clearly, I think we are talking again about quite different things here. And I may agree with you, for example, if an originator would change the cell line, if an originator would come up with a new transfection, would do then really come up with this product, you may think about a proposal like this. But this is a completely different story, and really one should be fair when making this proposal. If you do a manufacturing change and you can do actually a comparison to a certain manufacturing history. I think it's very clear also biosimilars, and eventually this gave the wrong impression yesterday, will introduce manufacturing changes and you will get exactly the same picture you have seen for the originator. biosimilar in that sense are not worse and are not better than the originator when it comes to comparability. But really starting a process, a new process from scratch, it's a different situation. And I think everybody is accepting this, including FDA, EMA, and so on. We are talking about something different here. And therefore, I think it's justified to say that you may really apply here new INN or whatever identifier. You even may think about sticking to the same INN, but having an additionally valid other identifier, you know, also might be a possibility. But I think we can discuss this afternoon.